you. So we have to wait or we have to start?
Just a minute, sir. I will just, I'm sharing slides. So, very good morning. Now we can start, I think, slowly.
Share screen, ma'am.
Okay, I'm sharing this screen.
SAR biography, yes.
Is it?
Ma'am, it is not in PowerPoint, not full mode.
Biography is not needed.
Yes.
Sir, just a small one, sir.
Because Ashokji knows, when a speaker will speak, they will come to know about him. Am I correct, Ashokji?
Is it visible?
Yes, ma'am. It is
Ravi?
visible, ma'am. Yes, ma'am. Ma'am, please continue.
Okay, so very good morning to one and all. So today we have in training program very learned and eminent speaker, Professor Arun Kumar Joshi. So sir is at present CIMMIT Asia Regional Representative and Managing Director, Borlaug Institute for South Asia. Regarding sir's education, so he has obtained his master's and PhD degree from Banaras Hindu University, the greatest institution of India. And he was the former professor of genetics and plant breeding at Banaras Hindu University. He has mentored several students. We know several of them around us, even at NBPGR also. And regarding sir's experience in research and teaching, so sir has more than 38 years of experience in research and teaching. And regarding development of varieties, so more than 5 dozen wheat varieties have been released with the contribution of sir in India as well as South Asia across different countries, and which is very much popular across different countries and which is contributing in the increased yield of wheat across the globe. Then sir has very much contributed in breeding wheat for climate resilient, mainly for heat and drought tolerance, then disease resistance also. In a spot blotch resistance, sir has the major contribution, then in conservation agriculture also, and recently as nutritional sustainability is very much important. So sir has played a great role in zinc-rich biofortification program in wheat. Then sir has been engaged in various collaborations globally on climate resilient agriculture as well as integrated food system. He has also educated thousands of stakeholders and farmers across the globe in public sectors. Now coming to the award, so sir has been bestowed with several awards, like he is the fellow of Indian National Science Academy, National Academy of Science in India, NASI, the National Academy of Agricultural Science. Sir has also received Genie Borlaug Lobby WIT Mentor Award from BGRI, Cornell, USA in 2014. Regarding unique germplasm registered, so sir has, to his credit, 12 unique germplasm material. Then he has also developed one patent. And regarding publication, so sir has published more than 188 research articles in renowned and internationally reputed journal. And then regarding several extension articles, manuals, and book chapters he has published. So I request sir to deliver his lecture in today's training program. And the topic of such lecture is wheat genetic resources for a food secure, climate resilient South Asia. So, sir, I request you to please deliver your lecture.
Okay. Thank you, Jyoti ji and Ravi ji for starting this session. And I can see Dr. Ashok Kumar.
Good morning, sir.
Good morning. So I was telling introduction is not required.
Yes.
Yes, sir. Not required because you are already known to everyone, sir. So certainly introduction is not required.
No. Not that way. The way I will speak, if I am able to tell them one something interesting, or I am able to feel them refresh. This should be our CV. And Dr. GP Singh, certainly, I'm grateful to him for including me in this series where I'm in touch with younger generation. So before I speak, go there. I am telling to all my young colleagues here that I have learned more from my students rather than my teachers. From teachers, I used to get blessing and knowledge from students. So I have learned more from my students rather than my teachers. Because they are always better. And if I don't know very good, if I know very good, so ultimately it's what we call a collaborative knowledge sharing. So thank you very much. And second point, if my colleagues here, young people, can keep their camera open, then that will be better for him. They can close also
Yeah,
because
I
some
request
of
all
you
the
may want.
participants
No,
to
no,
switch
don't
on the
worry.
camera.
Don't worry, Jyoti. I'm just telling because in classroom also some of them used to sleep. And this is a good teaching. If you are sleeping means very good teaching. Classroom is running very
Miss,
nicely.
अभी तो सर मॉर्निंग क्लास है, अभी तो नींद नहीं आएगी सर.
Then you are in peace of mind, actually. So, and many of young colleagues have a feeling, which I also used to have, that that they know a lot of things. This is correct. You know a lot of things, but our brain cells, neurons keep on, what I'm going to tell is nothing very complicated. Because basically, I'm a wheat breeder. I worked in seed system, nationally, internationally. So I just will try to refresh you a couple of things which you know. And maybe that I have forgotten something, you have forgotten something. So this is the way we will discuss it. So maybe when I will share my screen, I will not be able to see you. So if you have any question in between, please raise hand. And don't worry, you can raise hand anytime, anywhere. Sometime by mistake also. I will not mind it. Don't worry about that. So, okay, so let me go to, yes. So if a screen is coming, you have to tell me it is coming.
Yes, sir.
Yes, sir. Coming, sir.
But it is not in,
Presentation mode.
now
Yes.
is it okay?
Yes, now it
Yes,
is.
yes, sir. Yes, sir.
Sir, it is okay, sir.
Thank,
Yes.
thank you very much.
Yes.
Actually, I'm giving this talk from my home, not from office, because I believe that internet in my home is better than office. The reason is in the morning, everybody starts working and sometime, whole CG center, NAS complex. So that's why I'm here. So if you face any issue, please let me know. So dear friends, I have taken a very, very simple topic, conservation, exchange, and use of wheat genetic resources in the global context, but actually it is mostly regional, South Asia. And why I have taken this topic so that I may be able to tell you some examples from plant breeding, wheat breeding, to tell you how important is germplasm and how important it is to ensure our food security, livelihood, and it runs whole country. Now, it's not moving. What is the problem? Hmm,
Sir,
this is the problem.
slide पे double click करके then it may move, sir. Double click.
Yes, sir. Moving,
Yes,
sir.
yes. So, dear friends, I hope you know this man. If you know him, if you know about him, I'm very glad. And if you don't know about him, you should read about him as much as possible. And this is nobody but our very honorable Vavilov. He created the first gene bank of the world, in a way, 1887 to 1943. And I hope you know that he died on 26th January. 26th January is the date you will never forget. 1943, in a labor camp, actually in a prison. And he believed that whatever modern crops are there, domesticated, They are susceptible. They are susceptible to natural disasters because they don't have genetic diversity and they are not so hardy. So they cannot withstand hardships. And he made 115 expeditions to 64 countries, collecting 380,000 samples. So he was a remarkable man. And he worked in the present day Petersburg, which is earlier it was Leningrad, the Soviet Union's Bureau of Applied Botany. So if you happen to go there sometime in your life, please visit that museum. You will never forget, you will always realize, I will say, the importance of the germplasm conservation, its use, and how great this man was. And if you see that in the top, I cannot see this somehow. Sorry. Yes, this was published. Sorry, this was recently published in an article. It says it was the first seed bank of the world. Beautiful story. That how he fought with against people who were opposing genetics, the fundamentals of genetics. Anyway, that's a history. But he was associated with all crops, including cereals, wheat. Wheat was very important in Russia. If you remember, wheat became popular in USA because during World War, wheat supplies were stopped from Russian Federation.
हाँ बद्री प्रसाद चौधरी अंदर बुला रहे हैं पेशेंट का नाम बद्री प्रसाद चौधरी मेरे यहाँ पे बद्री प्रसाद के नाम स्पेशल
आज 2 लेक्चर हो रहे हैं क्या
Kindly mute yourself.
Please, dear participants.
So dear friends, don't worry. Sometime you get one free out of one lecture, you know. you go to market and something is free. So you are getting one more free lecture. So don't worry. Anyway, I was telling about Vavilov. So Vavilov is, amazing man. And another interesting thing which you know about wheat is wheat domestication, it has been said, is responsible for the increase in human population. See, this is very interesting. Many people say the population has increased because wheat became so successful. And it is true also, because when wheat production was not happening in Asia, including India, then many people forecasted that half of India will perish. So if you are working in wheat, rice is equally important, then please don't feel that you are being blamed for population increase. Rather, you are responsible for the happiness in the world. And here, the germplasm, genetic resources, they play the leading role. Now, just a simple, I read a newspaper which says the 10,000 year success story of wheat, and it has been published by D'Souza. It simply says that earlier it was emmer wheat somewhere before 10,000 BC. Some people are driving, they are late actually. Some people are driving, so they are still coming. So anyway, so about 7 to 8,000 BC, cultivated emmer came. It was durum, difficult to thresh. Then spelt came. Spelt is still grown a little bit somewhere. Then durum wheat came, and then bread wheat came, present day common wheat. Then there was green revolution, and now it is all increasing. So normally we eat triticum aestivum and little bit triticum durum. Some other wheats are also there. Now phylogeny, I am only showing you not to teach you the evolution of wheat. This is simply to tell you that alternatives, relationship. So there are in nature a number of species which are related by blood. And therefore, whenever we want a solution for something, we have to go back to history. For example, if you are in a problem, you are a young man, a young girl, first thing you do is you call your mommy and daddy. Papa, I am in problem. Mummy, what should I do? So these are papa mummies. So whenever we see some challenging issue of a disease or stress or climate change, something, we start investigating the history. So we will want to go to wild diploid Triticum urartu or spelled twice, or monococcum, dicoccoid, toshai or dicoccum spelled something, because crossing is easy here. You can transfer genes. Now, currently you have gene editing, but gene editing has a lot of limitations at present, maybe 10 to 20 years, because first you know about a gene. Where is the gene? So this is only to tell you how to connect with the history for present day solutions. And if you want to see the largest gene banks in the world, it is given in the book, several books you will find. This is the current data, that the largest gene banks for wheat, I'm just showing NPBGR. So National Bureau of Plant Genetic Resources is still quite large. So it is among top 10, and it is growing every day. It is collecting germplasm from different countries, from CIMMYT as well, where everything is free. So if you want any germplasm for an international institute like CIMMYT, it's free. Otherwise, you cannot get a single seed from anywhere in the world without an agreement, which is very strict agreement. But CG centers provide with a very simple agreement. So CIMMYT has the largest wheat collection. You can see here, 141,759. So these are the top ones in the world: Australia, China, Italy, Lebanon. But the name of India is there. This is a matter of great pride for us. Now, but in situ reserves are also present. So you may wonder how we can conserve germplasm in a grassland, in a farm, but actually it is true. There are locations and they have been named by a reserve, just like you have Kanha National Park for tigers. It is Arbuni, then there Ham, all those are there. You can see the name in different countries: Armenia, Israel, Lebanon, under Lebanon, Syria, Turkey. So as long as you will not see any construction activities or other activities going on, they are surviving and they conserve a lot of related species of wheat, different land races also. Now, several studies have proved that wheat accessions can be useful. Several studies have been done that accessions which we have in germplasm, for example, this was published a paper in 2020 that when diversity analysis of 80,000 wheat accessions was done, this was done in CIMMYT because it has a large collection and there is a project, then again, it's stopped. So my slide is stopping it, maybe because of internet. Yes, now it has come. Now, these 80,000 collections were hexaploid, 56,000, 18,000, or 19,000 tetraploid, and some wild. They used different types of modern technologies, which you are using at present. You know about DART, you know about how to find out SNPs, etc., etc. And they found that 72% of markers were unique and 50% had some connection with some gene. So many genes were known also, but the analysis revealed, it informed that land races are very useful still unexplored. There is diversity and in different regions. So you can know what are the reasons. You can see here, if it's very small here, but land races, they come mostly from Iran and Pakistan. Synthetic lines are there, modern land races from Mexico. So there are different countries and regions and different types of varieties. So they are useful. They are proving again and again, because tomorrow you may need something very interesting. Now, another interesting study, which was done recently, it has been published in 2022, the physical map of location of S1Ps were discovered for heat and drought tolerance. Dear friends, climate change means what? Climate change means heat and drought. And you cannot predict when it will happen. Whether it will go to, if it is going to minus, that is different. But if it is going to plus side, wheat loves winter. So wheat will suffer. So food supply will suffer. And you can see one war with Ukraine. Only one war between two countries has disrupted the food chain of the world, fertilizer chain of the world. So now investigators are very much interested in heat and drought tolerance. So they used 990 land races from 33 countries. And they mapped on different chromosomes useful genes, which we can use tomorrow. Now, please remember the 990 number is not a big number. You may believe that, oh, in NPBGR or my institute, I'm handling more number of lines. That's very good. You should. But what I want to say, again, it is stopping my, there is some problem with internet or something. So please bear with me. There is some problem. Next. Yeah. Now, so what they did, they collected from 33 countries, and a simple diagram you can see. They use some parameters, temperature, longitude, latitude, altitude, precipitation, etc. 7 parameters. There were a large number of genotypes. Certainly there was phenotyping. Certainly there was genotyping. Now GWAS, everyone is doing. And then they, based on data, they found that for drought and heat, you can get new genes. So, but actually, the research is not so simple. They screened actually 174,553 accessions. So these accessions were taken out from the gene bank. Then they filtered using their data, duplicates, etc., a lot of bioinformatics. Okay, 1151 land races of Triticum estimum with unique and sensible geographic coordinates. Yes, the data is making some sense. If a land race is surviving in a country for 50 years, 80 years, 100 years, there must be some reason. Then they did all kinds of location mapping, and they finally concluded 990 land races can be useful, can be validated with geographic data. Otherwise, same line may be present in different data sets, and you will not get a correct result. So accessions, I told you, came from 33 countries and distributed to 13 geographic regions. So doing this kind of work is very challenging. It requires a teamwork, but it has been achieved. Now, what is this picture? This picture is, don't neglect it. It is Agilov's neglecta. So you cannot neglect me, this, a wild wheat relative. So after every 20 years, you can see, where is the seed, how you will get seed. It is in -20, -30. You store for a long time, just like NPBGR. So approximately every 20 years, CIMMYT has to regenerate wild wheat relatives in greenhouses so that we can have healthy and viable seeds for future whenever necessary. And this picture has been taken just to tell you that plant breeding is, for plant breeders may be easy, but conserving germplasm is not so simple. Some people think, okay, take a packet, keep in the refrigerator, my job is done. Not like that. It's not so easy. Now, this is just to tell you and give you an idea that genetic resource research, conservation, utilization are supported by different units. You need a seed health unit. Seed health is very, very important. Quarantine, extremely important. You know why? Biometrics, so that you may know whether you have a correct data set available, correct germplasm available for your study. Statistics. So we have to, and data management. So all the time we have to have a beautiful team of classical scientists to lead to a good research. Now, CIMMYT is fortunate that there is a long-term project, Seeds of Discovery. SEED, it is called. So it is handling germplasm and it is studying biodiversity for maize and wheat both, so that we can use those lines, those genes for future breeding. Now, so this is for teamwork. Now, next, but genetic resources, a genetic resource has no value. If you have several resources, they have no value if it is not constantly used and improved seed shared with people who need them. Without that, it has no value. Now, if you see what you are doing in India, same thing we are doing in CIMMYT, that millions of improved maize and wheat seeds are sent annually from CIMMYT hubs, Colombia, China, Ethiopia, India, Kenya, Mexico, Turkey, and Zimbabwe. So CIMMYT annually on an average, we dispatch 1,500 shipments. One shipment means one courier, one speed post. It may carry large number of seed samples. These shipments contain 500,000 individual seed packets, 500,000. So you can see the logistic handlings carefully, without mixing, without disease, without contamination, right time, right place, right address is not easy, because it goes to about 100 countries. And there are about 800 recipients globally. For India, it will come to IIWBR through NPDGR. So massive work for use for future, and there are success stories. Now, what are those success stories? You know most of them. I believe that you know this man. And I believe that you know this man. Now, who are these people? Dr. Borlaug. And he is the man who produced Norin 10. Gonjiro Inazuka. He was in a Japanese research station, Iwate Prefecture. So 1935, this was registered, Norin 10, as a numbered cultivar by the ministry. And it was called Norcinso. Norcinho you may call, because in Mexican, you know, SHJH. So we call it Norin 10. It was number 10. And then it was given to the world by a biologist, and he was actually army officer, Cecil Salmon, who was with the General Douglas Army in Japan after Second World War, and he found, oh, wheat is so dwarf here. So they never saw the dwarf wheat. Although dwarf wheat was present in Korea, it was also present in Eastern European countries in different names. Rarely, because that time is for drought, and drought tall varieties were surviving. But in Japan, he collected 16 lines, and then the rest is the history. That how it happened, how this went to USA, and how it was used in winter wheat, and then from winter wheat it came to Dr. Borlaug, and then happened Green Revolution. So this is a beautiful success story. And Dr. Borlaug has said to Gonjiro Inazuka, without you, my research would never have been possible. So he offered him sincere thanks in one of his lectures and writings. And because of that, you can see this, what is happening and what has happened. You can see the world wheat. Now, this is Yaqui Valley, Mexico, because Dr. Borlaug was there. You can see the curve that every year there is increase in wheat productivity, and therefore in the production. The yield, you can say, is increasing. The rate is going down certainly, but this is extremely important, as we understood during COVID time, that 80 crore people were given free ration by government of India because of the buffer stock. It's a world record. So it would have never happened without that germplasm and the success story of the use of germplasm. Now, synthetic hexaploid, this is another success story, something new. Because when diversity was not enough, many people felt, then some people thought, why not to develop synthetic wheat? And CIMMYT established its White Cross program in 1986. So synthetic hexaploid, AABBD, is developed by artificially generating a fertile hybrid between a durum wheat tetraploid and diploid wild goat grass Agellos toshai. So they created for a certain purpose so that we can get more resistance against diseases, more tolerance to drought and heat, and also strong stem so that there is no lodging. So this program was initiated. And this paper has been published in 2019. There are two sections, left and right. Now, what is this? This is development of synthetic wheat, and the right hand is natural evolution of hexaploid wheat. So they tried to photocopy that in nature, evolution has occurred like this. Let us try like this. And after that, a number of synthetic wheats were developed. So please see, it started in 1986. Many of you were probably not born that time. And I started my assistant professorship in 1985. So now the results from international yield trial show, after about 40 years, that 20% of the advanced lines were synthetic derived with an average D contribution of 15.6%. This study came in 2-3 years ago, and they are maintaining and enhancing genetic diversity and genetic gain over years. So this is a beautiful example of creation of genetic diversity and its use in breeding program. And there is data based on SAVIT, which is for semi-arid wheat yield trial, very famous trial distributed throughout the world. Elite spring wheat trial, which is distributed throughout the world. And you can see it started in 1997. 1997, so after 10 years, when they started using it, it entered the breeding program, and then it remained slow, and then suddenly it is increasing, going down, going up. In ES-wheat, it is going up. So it means the efforts were very useful. The money spent is being used. Another thing, another talk is coming, dear friends, for you. So don't worry about the free talk. So another problem which has been faced in India, that early sown wheat is not successful. Farmers are advised to plant in November. Please do not plant in October, because the cause, what is the cause? The cause is that there is early heat. Temperature is higher in October. It is higher in middle October to third week is very high. Fourth week, little bit okay. So some farmers may plant in Punjab, Haryana on 30th October, but they cannot plant in 20th of October because there are no tolerance to early heat stress, and suitable varieties were not known or not developed. So what was the consequence? The consequences were wheat crop is denied its full yield potential due to shorter crop cycle. So when they plant late, the match becomes 20-20 rather than 50-50, or like a test match. And October, when you are not planting, then there is soil moisture because of monsoon. Because monsoon will withdraw around October, soil is still wet. But by the time you plant in November, it will dry. You have to give pre-sowing irrigation. But here, you can use it. And wheat crop is exposed to terminal heat stress in March, which happened last year. And you know the story, what happened last year. Within 3 days, we lost more than 3 million hectares. Some reports came, final report is that we lost about 3 million, 3.5 million. What happened? 1 minute. Okay, so what we did, how we used germplasm? Immediately we brought 3,324 advanced lines with diverse pedigree from Mexico and planted under October, third week of sowing in 6 locations of India, as well as in Mexico. Their heading days very varied. Global cultivar, you can see how much they were varying according to location. It was Ludhiana, it was Karnal, so different locations were there. And they were varying for different trades. Then what happened? Then we found, oh, early heat tolerance is present. We had no idea. We did not know that there was juvenile heat tolerance present. So everything's present in nature, in your breeding stocks, in your gene bank, but we don't know. So this paper was published telling us that this is possible. And October sowing proved that by simple change, there is around 1 ton per hectare yield advantage. You go to 20th October, 24th October, instead of first week of November, and 1 ton you will get same resources, less water, 1 water less because you are using moisture, and you can see the early sown. So when we use 540 lines early and normal repeatedly, we found that early sown was giving higher yield. One more free. Please mute, dear friends. Some people are driving car, scooter anyway in the field. Anyway, so they are not interested in lecture, doesn't matter. So early sown. So this is how we prove. You cannot prove by 1 line, 2 line, 3 line, 1 crop season, 1 location. So when it was done in 6 locations repeatedly, and when data of Northwest Plain Zone was collected, there were 3 locations that time, 3 were in central India, then it was proved. So this is how we can prove. So 3 varieties were released in 2020 and half a dozen more. Jyoti ji, can you give them instruction?
Trying to mute, sir.
So that although their voice is very sweet, but anyway, so dear friend, next question. What will you do when a new disease will come? A virulence will come, like COVID-19. What you will do? You will develop a vaccine. Immediately, you try to throw alcohol, you try to throw sanitizer everywhere. You will take a bath of sanitizer sometime by spray. So when suddenly a new disease comes, a virulence will come, then there's panic. Shares will go down. There is sense of food insecurity. So simplest thing is collect all available advanced lines, whatever available, wherever, use money, and you have to screen at the location where the disease is present. If a disease is coming in Africa, you have to go to Africa. If it is coming in Latin America, you have to go to Latin America. You cannot test here. Why? Why it is done? It is done simply because seeds of resistant varieties are like a vaccine. Seeds of resistant varieties are like a vaccine. And I wrote with Hans Braun, the former director, a book chapter in seed. It's a simple chapter of seed for textbook type. But there we thought like that after internal discussion, where the global program director, Alison Bentley, was also present, that what is a resistant seed? It is like a vaccine. So it is necessary to find out a resistant variety through large-scale screening. Now, there are several examples of successful example. This is UG99. When it was coming, many people thought it will not come. Like COVID-19, it will not come. It will not come from China. It will not, new race will not come. And what happened in India? What happened in the whole world? And now China is suffering because of COVID-19. But this UG99 was stopped by intelligent investment. Invest, produce, distribute, fast-track manner, seed of resistant varieties. These are some examples. And India played a very, very important role. You can see here, it's a huge area. This year, wheat is in 34 million hectares. 34 million hectares. Some countries don't have the area which India increased in 1 year. So you can see, and another is wheat blast. You can see one is blasted, other is normal. It was in Latin America. Now it has come to Bangladesh. In 2016, there was a panic. So same story was repeated. Same methodology was repeated. Advanced lines were twisted. Where? In Bolivia and Bangladesh. So this is Bolivia. You can see Okinawa and Kurasila. They are Japanese names because Japanese soldiers used to live there. It was Japanese colony, and this is Bangladesh. So we followed same technology, and first variety BAW 1260 was released next year. Now, many people think how you can release next year. Same methodology. We sent hundreds and thousands of lines in Bolivia and Bangladesh tested, and we found advanced lines which are ready to release carry resistance, and that was Bari Gom 33. Bari Gom is wheat, Gehu 33, and it is also biofortified. So this is a success story, and large-scale seed dissemination was done. And since then, India has released a dozen wheat plus varieties more. Dr. G.P. Singh can tell. I am telling you very conservative figure. And more than that are in pipeline because now zinc is this wheat blast is extremely important for release of varieties. Now, a policy has been made by IIWBR ICR that if there is blast resistance, we will give variety a priority. So it has a priority pass for boarding. Now, another successful example is biofortification, genetic. Many people believe you cannot increase zinc grain in the grain. It has to be in the soil. You cannot increase more than 2%, 3%. Or maybe by fertilizer you can do something, but genetic is not possible. But then a search was made because not a single cultivated variety, whether in the field or in the gene bank, had high zinc. So they used synthetic derived estimum, wild relatives, land races, and did a survey, and then found that, yes, we can get some lines, cross them, pre-breeding was done. So this was pre-breeding and best lines were deployed. And now you know the history that zinc content is very high along with grain yield as well. So zinc content, zinc concentration, soil is also equally important. So ultimately we found that yes, it is possible to find out wheat lines which have capacity to absorb more zinc and carry it to the grain. So there are large number of examples. Now land are very important. This is example of Middle East beyond Afghanistan, Turkey. There is terrible earthquake. I'm sorry for that. So very bad news. So you can go to Uzbekistan. So these are the different spots. So we selected about different selections, 7 in 2 in Afghanistan, the different countries. And then a study was done. We found that yes, we can group them. Land races and modern germplasm, they fall in different categories. And then we found that under irrigated conditions, modern germplasms are better, but not under rain-fed conditions. So no significant difference noted Afghanistan and Iran in 2019 under rain-fed conditions. So rain-fed conditions, land races have higher yield than modern germplasm. when there was a severe drought in Turkey in 2018. So you know the benefits, but we are proving it that yes, these are the lines which are performing. So there are some genes, we can use them. So this is all indication, and which we all know about it, that yes, drought tolerance is present there, and in future we can have better lines. And this is about nitrification. What is this? This is something new, nitrification. How to stop nitrification? If you apply urea, it is lost, 70% lost. So there is, you can see, Lemus racemosus, there is a wild relative, so BNI, biological nitrogen inhibition. So this is normal Munal variety. Munal is released in Nepal. In Mexico, it's very high yielding line. And it was also released by a private sector in India, if I remember. But you can see in 3B chromosome, you can have a segment from racemosus. So what is the benefit of this? That this now pre-bred source is ready now. Already there is a project. We are already working with IRI, IWBR, and Salinity Institute in this project. So the production and release of nitrification inhibitors from plant roots is termed biological nitrification inhibition. So roots produce something so that if you apply ammonia, it will remain as ammonia. It will not get converted into nitrous oxide or something. It will not be released into the atmosphere. So slow release is natural phenomenon. Very interesting story how it happened. But a chromosome reason is there which controls BNI production in wheatgrass, Lemus racemosus. And now we have crossed it, and high yielding lines are already there. We are making crosses with even better lines, and we want to release them as soon as possible. ICR, I'm thankful to ICR for this project, and BNI Munal and BNI control. You can see the advantage. Total nitrogen uptake by shoot and grain is much higher if there is BNI. So nitrogen use efficiency can increase by 30% by this. This is a beautiful research. And if it is successful, it will save 30% fertilizer of government of India and many other countries. So this is another beautiful use of genetic resources. Now, couple of slides because it is already 10:21, although we started a little late, so that if there are some questions. So 1B1R, the Weary lines created history by sustaining green revolution in 80s. But we later on found that 1B1R leads to poor quality. Quality is not so good. I mean, it's an impediment to improving the breed quality. So the breeding program was so arranged in CIMMYT that 1995, it was almost 70-80%. Everywhere it was, frequency was everywhere, more than 80%, 90%. Now it is close to zero. So this is how through an organized breeding program, you can increase or decrease the frequency of a particular gene or a type of trait you want or you don't want. Just like malaria, now there are gene editing is being done in mosquito to increase the number of males, not females, because you are attacked by all the time female mosquito because of egg production. It requires protein. Your blood is the best source of protein. Male can get food anywhere. So that kind of research in gene editing is being done. I'm just giving an example because it's not only about wheat, it can be anything. To speed up genetic gain, technologies are changing, so tomorrow will be different. What I have told is different. Tomorrow, gene editing, something magical may happen. Maybe you will get edited by yourself. Nobody knows. And if you see how crop breeding progressed, I have taken it from this slide from somewhere, and I was very much happy to see that. You can see here, up to 1900, 10,000 years, we were here. And within 100 years, we are suddenly moving so fast. So many technologies are coming so rapidly. So technology is changing the world. So in 12,000 years, we were in the same type of research, selection, crossing, some selection, some crossing. And suddenly everything has changed in 100 years. And so science will change very, very fast. So my dear young colleagues, you have to be very prompt in bringing innovation. And don't think your lab today will be useful tomorrow. Try to outsource, make collaborations, but genuine. Please do not do anything which is not genuine and not in haste. Good planning is required. So there is considerable scope for regional global collaboration in conservation, exchange, use of wheat germplasm, all crops actually. I give example of wheat just to focus. And it will be more important under climate change and growing stresses. Stresses are increasing. Shivering, New York is shivering. There is Arctic blast there. And what happened here? Suddenly, temperature increased in India. So we have to be ready for all those things. And I just want to, people who respect to all of you who work on conservation and use of wheat germplasm. This is just a picture because Tim Penn was leading this program in CIMMYT for more than 30 years. Now he retired. So he was at the Global Seed Vault in Svalbard, Norway, for the official opening ceremony in 2008. And he's holding one of the seed boxes used to store nearly 50,000 unique maize and wheat seeds collections deposited by CIMMYT. I think India is also contributing. And one day you may want to go there and see that how important is that place. And with this, dear friends, I thank you all for your interest. And I want to stop sharing here and I'm in your command, please. Thank you.
Thank you, sir. And I would like to request participants to ask their query.
Yes, if you have any
Yes, sir.
question, if I was wrong somewhere or you want to add something, you can write also your question. I can see from here chat box. I think there are some questions. No, not here. Okay. It was please keep yourself muted. Now you are not requested to be muted, please. So according to time schedule, 4 minutes are left because how much up to 10:30 you said, Jyoti?
10:45.
10:45.
Okay.
We have time, sir. We
So
have time.
if you will not ask question, I will ask question.
ठीक है, sir. You can ask.
So somebody's so who is working? Anybody working in wheat? Can you raise your hand?
Yes, sir. Some are there, sir.
I'm not seeing any hand.
Yes, Dr. Sombir, are you there?
So then he should be in the wheat field with mobile, listening the lecture.
Yes, ma'am. Yes, ma'am. I'm
Yes,
here.
sir.
Sombir is here. So Sombir, you are working in wheat. Okay. Is there
Yes,
anyone?
Yes, sir.
Sombir, introduce करिए
sir,
sir को.
sir, I am Dr. Sombir Singh. I am from CSE University Kanpur. I met many times in wheat workshop, sir.
Very good, Sombir ji. You are in Kanpur.
Sir,
Okay.
yes, sir. Yes, sir. When we release the variety K1317,
चलो जाओ तुम लोग, अब हमारा काम हो गया, हमारी मीटिंग चल रही
by जी sir.
K9107.
1317.
Oh, 1317. Yes, Kanpur is famous for Kanpur wheats. KW. So at one time when I was young like you, I was in, even when I was assistant professor, whenever I used to go to purchase wheat, I used to ask a shopper, dear friend, what is this variety? He will sell, he will used to tell me, sir, KW wheat hai ye, KW. मैंने कहा KW क्या होता है यार, ये बताओ मुझे. बोला नहीं sir, all good varieties are called KW. And then they become Maulvi. because then HUW 234, nickname Malviya 234, became very popular in Eastern India. So many people started calling it Malvi. Some used to call it Maulvi. So when you ask anything, it is Maulvi. Malvi, sir. So Kanpur, you are from a very prestigious place. Now, one question I can see somebody is
Thank you,
asking.
sir.
Hello,
Thank
sir. Hello, sir.
हाँ विनोद जी.
Sir, myself, Dr. Vinod Kumar, sir. I am beat breeder at Pawar Kheda Center.
Oh, thank you.
And
Very
I
good.
am in, sir, field. I saw my field, sir.
I know you are in field.
This is, sir,
Yes,
segregating
please.
generations.
Ah, beautiful.
And, sir, this is cement nurseries.
Wow, 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 wow. Very good. Very good.
This is nurseries, sir.
Very good.
We have three,
So,
sir, SAWYT, or all three nurseries we have got, sir, from cement.
very good. So, can you show your field? Yeah, please continue to show your field one more time because
Yes, sir.
next,
Yes, sir.
Live
because
demonstration of
no,
Yes, sir.
because
field.
Jyoti told me that this will not be very interactive. This is like classroom lecture, but this is not a classroom lecture.
Yes,
Very
sir.
much interacting, sir. No.
Yes.
We can see field also.
And
So
sir,
I
I
can,
want to show the germplasm bank, sir. We have, sir, more than 6,000 germplasm.
oh, yeah, I have seen
And
your,
our
yes.
previous leader, sir, Dr. P.C. Misra, sir, he is retired last year, sir.
Hmm. Yes, yes, I have visited to your station. I have seen
Yes,
your
sir.
germplasm collection. Pawar Kheda is very, very important center. But dear friend, there is a question from, thank you. There is a question from JNKBB who is asking this new now heat is a major limiting factor for yield and quality of wheat. What will be strategy to develop suitable variety for Madhya Pradesh?
Dr. D.K. Payasi,
This is,
sir. Hmm.
sir, this is germplasm genetic stock, sir. And this
Hmm.
is let's on trial of ICRP, sir.
Thank you, Dr.
IVT and
Vinod.
IVT.
Now, sir,
Okay,
will
ma'am,
take up
okay.
another question.
No, actually Monu has also raised hand and
Yeah,
one
Monu
question
has
is
also
here.
raised his hand, sir.
So first I should answer this one. JNKVB, who is asking?
Dr. D.K. Payasi,
Dr.
sir.
D.K. Payasi, myself Dr. D.K. Payasi.
हाँ जी
I'm,
बताइए.
sir, heat is limiting factor and affected seriously in yield.
Hmm.
Yield and even 100 seed weight of the crop. Then what will be the strategy to develop suitable heat tolerant variety for Madhya Pradesh, sir? Because farmers are now worried about the heat tolerance. In February itself, the temperature goes high, and it needs.
Hmm.
Yes, sir.
I think this is the question being
I am
asked
from
by.
Sagar, sir. I am from Sagar, and Sagar is one of the ICRP center for rainfed breeding, especially.
Acha, you are in Sagar. Okay,
Yes,
okay,
that's why I am asking.
okay, okay. Thank you very much. Actually, this question is being asked by almost everyone every day. I ask to myself, what should I do? Because this is for all of you to understand the global modeling by climate experts of climate change. The modeling says that world average temperature will increase by at least 2 degrees centigrade if we don't do anything. So now we are working for wheat which can survive 2 degree higher temperature, 2 degrees centigrade. Now,
Yes.
now one is that breeding is an automatic process. When you start using your breeding material in hot spots, automatic some selection keeps on occurring. So in breeding, the most important thing is making intelligent crosses. So people say that make smart crosses. When we mentioned smart crosses, then our former director Hans Braun said, do you think the previous breeders were not smart? They were also very smart. But now you have to be smarter with respect to the present challenges. Those days were different. So first, please try to identify the best germplasm and the best cultivar. So don't try to use any cultivar anytime. No, always use your best performing cultivar, which is popular among farmers. That will be your standard. Then cross with a number of those lines which, according to data, have proven that they have heat tolerance. Then you cross it and then keep on doing the normal process under late sown condition or early sown condition, depending upon what type of heat you are interested in. At the same time, I will request you to collaborate with molecular breeders, because molecular breeders can speed up things using gene editing in 10 years. Nobody knows. Gene editing is moving very, very, very fast. So once you know that these are the lines which can perform well in high temperature, then you have to do some molecular studies side by side so that you may know about genes. You can map them through collaboration. Breeder cannot do alone. So you will continue to do breeding short term. At the same time, you will continue to do molecular research so that you can find out genes and gene editing people. So all these things will have to combine in next 10 years. Only breeding will not be enough. Only molecular will not be enough. So please do collaborations. So all kind of collaborations. So this is what you should do, in my view. Thank you very much. Now, Monu, your hand was raised. Sorry.
Yes, Monu, now you can.
Sir, pranam, sir.
Yes,
Sir.
Namaste.
Sir, I am from IRI Jharkhand, sir.
Very good.
Sir, I have started wheat research just last 2 years back. With the objective of cropping system and fortification. But at the same time, sir, I am facing another problem here. Sir, our land is characterized with acidic. Plant performance is not so good like that what we see in the Banaras and Samastipur region, sir. So it is a very big challenge for us, sir, here. And we have to give 7 to 8 irrigation to sustain the crop. So, sir, it is another problem for us, sir. So,
Monu ji, thank you very much. See, the world is full of problems. Problem, but still there is happiness. Your director is now from Maize. Am I correct? Sujay is your Dr. Surya is your director?
No, sir. No, sir. Dr.
Who
A.K.
is your
Singh, sir.
director?
IRI Delhi, sir.
Oh, you are in a Jharkhand different institute then. Sorry.
Yes,
No,
sir.
I got confused.
IB Ranchi, sir.
Sorry. Okay, so Monu ji.
Yes, sir.
Actually, I can tell you that same formula will apply. Breeding fundamentals are same. But if you are giving so many irrigations, then I will suggest you that agronomy is equally important. So you please visit BISA Center, Borlaug Institute of South Asia. I think should be in IRI, also other institutions, and see how we are using surface and subsurface drip irrigation to save water. If you are doing 7, 8 irrigations, first you switch to drip and subsurface drip for your research. In cereals, I am telling wheat, maize, rice. So you are in wheat. Then the same fundamental will apply. That you have to screen, go search global data, you write mail to us, we will provide you. You have to characterize your environment properly. That this is the type of soil, this is the pH, this is the average temperature, this is the rainfall, this is whatever it is, relative humidity, etc. And then you need this suitable line for this. So certainly CIMMYT will be happy to provide you a collection of germplasm which are performing well in other parts of the world under similar situation. It may not be exactly. Then you have to start a crossing program using the popular varieties of that area, and you will be successful. You will become a hero after some time. Jyoti will give you an award after some time with Gautamji. You know, Gautamji, I can see him. Okay, Monuji?
Okay, sir. Thank you.
Oh, you are in IRI. Is such a prestigious, place, IRI. So, Jharkhand, very good. So please, this is the process and you can discuss with me later on.
No, sir, Dr. Amit Gautam from ICARDA. He want to ask. So, Amit,
Amit
yeah.
Gautam?
Yeah, he has asked Amit Gautam from ICARDA, sir. He's also
Gautamji.
one of the training. Amit, if you are, you can ask yourself. Otherwise, from chat, I will read out. Sir, he's asking, my question is zinc and iron depends on soil and location. If any light wheat line is planted at different location, zinc and iron may vary. Then how to overcome that? Because if anyone claim for zinc and iron, that may differ. All.
Yes, it happens everywhere. You give farmer a variety, very good variety, 7 ton, and he will come, sir, it gave only 3 ton, 4 ton. So same thing in zinc and iron, So actually, from where the zinc will come? It will come from the soil. Even if you have 30% more ability to absorb, and zinc is a nutrient which doesn't move. It will not move. Zinc is not nitrogen that move, and it's not like you can move anywhere. Some people were moving during lecture also, so interestingly. So zinc cannot move. So this problem is very genuine, that it varies from soil and location. So what you have to do is you have to breed for specific locations, for specific environments, specific soil type also, and then you have to educate farmers that the result will be like this. However, if it is a rice wheat cropping system, then in rice all farmers apply zinc, and because they apply zinc and because they apply regularly in the rice wheat cropping system fields, zinc is almost uniformly present in the all parts of a small block. This problem will not be there, but it may happen in other places. So you have to tell farmers. Sometimes many people, they do soaking of seed, use agronomic techniques also. You have to combine a good variety with some agronomy for more consistency, then your claims will be better. But simple technique, very simple technique. It can be seed treatment, or it can be application of zinc carrying fertilizer in the soil uniformly. This is the only solution. Thank you very much.
In chat box, sir, one message from Dr. Tarak Nath Saha. You're a student during UG time at BHU. Thanks.
Very good.
So he has written, sir, thank you very much, sir, for nice information. I'm working on flower crops, chrysanthemum, tuberose, etc. Here also high temperature affect the yield and quality of the produce. I remember you taught us during our UG time. So nice to see you after a long time. So, if you are
if you are happy to see me, I am equally happy to see you after a long time, my dear friend. And if you are something which I taught you, I hope I did not, disturb your basic fundamentals, But dear friend, plant breeding is so interesting. It is evolution. Now, what is evolution? Now, evolution, it requires involvement. Unless you are involved, no evolution will occur. So if plant breeding is evolution, so first you have to involve yourself very deeply with your plants, see how they are suffering and how they are not suffering. When there's more organic matter, they are not suffering. Less organic matter, they are suffering. Same temperature. It means there is something. And there may be couple of lines, couple of varieties, like you are working on chrysanthemum tuberose, like tulip. When I went to Netherlands, I found there are 4,000 varieties of tulip 20 years ago. 4,000. So if you have 4,000 lines of chrysanthemum, something 5,000, plant there. Plant under little bit harsher. Plant such that they match that difficult period. So you have to do some modeling. When should I plant? so that it will match. Wheat, when high temperature problem started emerging, we started planting that. Our distinguished scientist Ravi Singh started planting wheat in 18th of February. Today is 7th February. They will plant after 10 days, same set of trial which they have planted in November, which they have planted in December. Big trial, huge trial. And also they started using agronomy. Never forget agronomy. When you are suffering from high temperature, when you are breeding for high temperature, please use agronomy. Drip, subsurface drip, 10% water, and no reduction in yield, even if temperature goes up by 5 degrees. These are very amazing. So it is all about collaboration. Never one research will help you. And I'm glad that you are working on flower crops. You will be always in high demand, because whenever we used to, sorry, 1 minute is there. So Jyoti, whenever we used to organize Kisan Mela, always we used to decorate the entrance with flowers in pots. So all, aristocrats, श्रीमती लोग जो आती थी, बुरा मत मानिएगा महिलाओं यहाँ पे, जो डॉक्टर्स की वाइफ, इंजीनियर वाइफ, वो गेहूं धान सब छोड़ के pulses, they used to leave everything. Oh, very beautiful flower, from where I can get this variety? So no question on wheat, rice, and pulses, etc. So you are in a very nice field, and I congratulate you that you will continue to attract attention. Thank you very much.
So I think now there is no question in chat box. So even if you have further questions, so you can write to sir. We'll be circulating the contact number and the email ID of sir, so you can ask further. So thank you very much,
Thank
sir.
you. My email, anybody can write, contact me anytime. I respond to each and every mail, not Yahoo mail, CG mail,
CG mail.
till I am not retired. After that, Yahoo mail. Thank you very much.
Hello.
Okay.
Hello.
Yeah, anyone?
हाँ, डॉक्टर अच्छी मीना मैम.
हाँ हाँ, yeah, बताइए.
Good morning, sir. Thank you. Nice delivery lecture, sir. I want to ask one question. Sir, I am working in Power Millet. There in the Jodhpur, there is the terminal stress in the early in the March in wheat. So what are the physiological parameters we can identify for the terminal stress in early in the March?
Dear friend, as a breeder, I can tell you, you can publish paper by physiological parameters. There are several physiological parameters. The most common is the temperature on the leaf. We use infrared thermometer, and we have found that there is correlation of that temperature with the yield. So if there is cooling effect, if, for example, some of young people here can ride bicycle in 45-degree temperature and go to see it. When we were young, we used to go by bicycle in 45-degree, 44-degree, 43-degree to see a cinema. No effect.
Yes, sir. Yeah.
Our genes were like that. And now if we go in 38-degree without a cap, we will need 4 glasses of water. So dear friend, the cooling effect is there. But cooling effect will be where there is water in the soil. So under irrigated condition, that infrared thermometer can tell you the cooling and it will help. But if it is a rain-fed, then still it may work, but not so significantly. And another thing is night temperature. Please measure night temperature. Wherever night temperatures are lower and soil is healthy, you get 7-ton yield, like in Jabalpur. In 124 days, we get 10-ton yield because soil is black cotton and nights are cooler. Days are not so cool. So please study all that parameter. First, define your environment and then go for this cooling, but always correlate with the grain yield. There may be stay green trait. Many people say stay green trait, but it doesn't correlate with higher yield all the time. So there may be other for theoretical importance, but this is, I am telling you from the practical point of view, never forget grain yield, never forget that leaf temperature, cooling effect, and you may see stay green trait also to some extent if it correlates with grain yield. This is, and there are large number of traits then you can study, because publishing paper is equally important. But first understand your environment, day temperature, night temperature, soil type, humidity, all those things. Thank you.
Thank you, sir, for your wonderful and interesting talk and making the session interactive. So Dr. R.K. Gautam, our head, he wants to thank you. Sir Gautam, sir, please, sir.
Namaskar, sir. Namaskar. Good morning. I think it was a very enriching and inspiring experience to have listened to Professor A.K. Joshi ji. And on behalf of entire organizing committee and on my personal behalf, I take this opportunity to express our sincere gratitude to Professor A.K. Joshi. I am very sure all the trainees have been richly benefited by your precious and practically useful lecture, sir. Not only benefited, but I think all are deeply inspired by your wisdom, vast experience, and great expertise. Sir, we all thank you very much and look forward to you for your kind guidance and support in our all future endeavors. Thank you so much, sir. Namaskar, sir.
Thank you. Namaste to everyone. See you. Bye-bye.
Thank you, sir.
Thank you, sir.
Thank you. Thank you, Gautam ji. Thank you.
Ma'am, next lecture is 11 o'clock, ma'am. Dear trainees, please be in online, and next lecture will start at 11 o'clock. Let me introduce Dr. Ajay Kumar Roy. He is a principal scientist and project coordinator forage crops. He is working in IGFRA Jhansi. And he is an outstanding scientist in forage crop improvement, biodiversity, biotechnology, apomixis, abiotic stress tolerance, genetics, silvihorticulture, rangeland grasses and legumes. And he is an expert in multidisciplinary subjects, as he has vast contributions in forage crop improvement and forage genetic resources. And he is a recipient of INSA, NAS Fellow, and ICR prestigious Rafi Ahmad Kidwai Award.
May I intervene?
Yes, sir.
I am not INSA fellow. I am only
yes, sir. Yes, sir. Yes, sir.
It is somewhat mistake.
Yes.
I'm an NAS fellow, not INSA. Okay.
Yes, sir. Sorry, sir. Yes.
Okay.
And
okay.
ICR Team Award for outstanding multidisciplinary research in 2007-8. And Dr. S.K. Vasal Award for efficient utilization of PGR by ISPGR, IJPGR, and NAIP Certificate of Appreciation for outstanding contribution on sustainable rural livelihood security. And next, Dr. K.A. Shankar Narayan Award presented by ICR Kazri Jodhpur for scientific contribution in arid zone research during 2018. And he is having a lot of awards from Range Management Society of India and International Technical Cooperation Training Award by British Council Division for research on molecular aspects of PGR at in Institute of Grassland and Environment Research, Aberdeen and UK. And that is a project of DBT Oversight Associates. And is none, actually I could not mention the lot of awards, but even I given very glimpse of awards. And he is having varieties. Total 19 varieties are there, out of which 15 notified by the CVRC and 4 varieties identified by BSc. And he is having a 25 unique germplasm materials which are registered with NBPGR and INGR. And beside all these things, he is a best teacher and he guided as a mentor, and around 9 PhD scholars and MSc 10 students are there. And his publications are more than 146 research articles, and other publications also 100. And even personally, I am very impressed about his book is that is minimal descriptors of forage crops. It is a very good work, sir. I studied that one. And sir, it is great honor to me. I am welcoming you, sir. Please kindly share your expertise and vast contributions, and please give your presentations. Welcome, sir. Welcome. Sir,
Thank you. Thank you for giving Ravi for this introduction. So I think the participants are mostly
Sir, participants
belonging to the teaching and
research, sir. Yes, sir.
Yes.
Sir,
So is screen sharing is enabled?
not yet, sir.
No,
Sir,
I think it is enabled.
share screen. Yes.
Can you see the screen now?
Sir, but it is not in double presentation mode, sir. It is short.
Is it okay now?
Yes, sir. Okay, sir. Okay. Yes, sir. Please.
Is it now okay?
Yes, sir. Okay, sir. Okay.
So can I start the?
Yes, sir. Yes, sir. Sure, sir. Sure.
It is about how much time allotted?
Sir,
45 to 50 minutes of 1 hour.
Sir, 1 hour for 15 minutes, sir.
No, there will be interaction also, I think.
Yes, sir. 1 hour lecture, sir, and 10 minutes you have interact, sir. Any students are interested.
Okay, I will then start.
Yes, sir. Sir, for your kind information, these are trainee young participants from ICR research institutes and state universities also, sir. And they are from 18 organizations, sir.
Okay.
Yes. Thank you, sir. Please continue, sir.
So now I'm starting it.
Yes, sir. Sir, it is in short mode, sir. Yes.
Is it okay? Okay.
Now? Okay, sir. Yes. Okay, sir.
Okay.
Yes, sir. Okay, sir. Please.
Okay. So my friends, I am A.K. Roy, and already my brief introduction has been given. For the last 30 years, I am working on various aspects of forage crops. So the topic of my today's talk is forage genetic resources and utilization in Indian context. Okay. So my actually lecture schedule is divided in 5 groups. First part will be introduction, then forage research network in India, forage crop improvement issues and strategies, IGFRI research in forage PGR, and a few success examples how we can utilize the PGR. So actually, I will say I'm giving an introduction in detail because most of you are not aware with the forage. I'm sure. Forage is such an area where most of the agriculture scientists are not even aware of the names and nature and other things. So I will spend some time on giving an introduction. Why forage? And where, how, and when we should work on forage. Okay. So now coming to Indian scenario, if you see in brief, in India, agriculture has traditionally been mixed farming orientation. When we say mixed farming orientation, it means combining crop production with livestock rearing. So livestock rearing is an integrated component of Indian agriculture, if we say, since the dawn of civilization. Or if we say that civilization developed with the livestock first. It is the man who first used the livestock, domesticated the livestock, and started with grazing them. So our civilization actually developed near the river or the water bodies. If we say all our ancient civilization, whether it is the Indus Valley civilization, whether it is the Egyptian Nile River civilization, all the ancient civilizations developed near the water source where the livestock were domesticated and man started living in a social life. So it is since the civilization, we say animal husbandry or the livestock rearing is our integral part. And in today's context, if we say it is a boon for farmers with small land holdings, but where the land is not sufficient to meet the livelihood, it is the livestock which meets the livelihood sources. So in India, still we say more than 50% or 70% of our livestock is reared on the rangeland grazing. More than 75% or 80% in the small ruminants like goat and sheep, and 50% in case of cows, we are still dependent on the rangeland or grassland or pasture land where animals freely graze and not cultivated. So for in other crops, you will say it is the mainly cultivated crops. Here we are talking on both cultivated fodder as well as the grassland range resources, where in the grassland the grasses or the legumes grow, which are consumed by the livestock, by the sheep, goat, cattle, buffaloes, and they are important. So it is quite different from the other crops where we talk mainly on the cultivated crop, cultivation practice, but here it is different. So if we say in the Indian scenario, again in India, forages are vital constituents of unique integrated system. I have already said. And an additional point is that it is a diversified, rich cultural environment. So we know India is a diversified country. We have different cultures, we have different climate, we have different situations. And so, along with the different cultural environment, we also have different livestock breeds. Livestock breeds in different parts have different demand, and so different fodder crops are suitable for different areas. So again, you see, it is not a single crop which can be grown everywhere. It is the different forage crop for different situation. So when we call it forage, what is the forage? It comprises of grass, legume, and other herbaceous species. Trees and shrubs are also important animal food, especially we say in arid, semi-arid, and hill condition. And we say in the arid, like in the desert or semi-arid area, Rajasthan, Madhya Pradesh, Gujarat, or the hills, it is the tree and shrubs which are more important component of the livestock forage. So when we talk of forage PGR, we also include all these in the list. So most forage grow in grassland, are produced by fodder crops and provided by fodder crop residues. So we say forage has three components. One is the forage fodder crop residues, like the maize, stover, and sorghum, bajra, stubbles. Then the grasslands have got different grasses, legumes which are consumed, also the trees and shrubs, and then the cultivated fodder crops. All these three components we say are important as the forage, and when we talk of forage PGR, we have to talk on all these three items. So why so forage crops is important? Because we have only 2.3% of the global land, but we have to support nearly 16 to 20% of world livestock. We have to support nearly 17% of human population. And mostly our farmers are small and marginal holders, where livestock is their main source of livelihood. So in this context, the importance of forage crops is much more higher than what we perceive or what we understood. And we say the cultivation of forage crop, it is nearly 4.8 to 5% of the total area, total arable land of the country. And it is stagnant for the last 3 or 4 decades, if we say. because the cultivable land is not spared by the farmers for solely for the forage, except in the hilly areas or in the peri-urban areas, where the fodder is very costly and it is in high demand. So when we say of forage crops, we face certain questions, certain unique problem associated with forage crops. One, I have already talked, multiplicity of the crop species. Because the grasses, the range legumes mixtures, they differ in different areas. It is not one grass which is suitable for all the areas. Arid zone have got its own grasses. Hill zone have got its own grass legume. Similarly, the coastal or the humid part has its own. So we have to work on the many crops across the country to make the forage crop deficit complete. And then the preference varies from season to season, locality to locality, and it is coupled with non-commercial nature of the forage crops. When I say non-commercial nature, why we call it non-commercial? Because it is still not directly being sold except in the peri-urban areas or hill areas I mentioned. That fodder crop is not directly sold. So to some of the examples, I will say that when we were conducting some experiments under the NAIP program on the Sustainable Livelihood Development Program, we simply asked villagers that what are your opinion about growing these fodder crops which you have grown for 1 or 2 years. And their comment was very simple. And similarly, I said, if I say in Hindi, साहब आपका ये घास तो बहुत अच्छा है, इसको लगाने से बहुत अच्छा हुआ, एनिमल का भी यील्ड अच्छा हो गया और दूध भी हमारा 2 किलो से 3 किलो 3.5 किलो हो गया, लेकिन उससे फायदा नहीं हुआ कि 2 किलो दूध भी बच्चे पी जाते थे, 4 किलो भी बच्चे पी गए, हमको प्रॉफिट नहीं हुआ. 1 किलो गेहूं बो दिया होता तो मुझे 1500 रुपये मिल जाता. So this is that if I translate in English, simply said, milk production increased, animal health increased, but it was not converted into a commercial entity because he was not selling milk. So if he is selling milk, it would have been profitable. Milk was consumed in his family, and so he could not understand its profit because the health was improved, but it was not converted into the monetary terms. Similarly, they allocate their most degraded and marginal lands for forage production with minimum inputs. A farmer will give regular irrigation and fertilizer along with the pesticide spray to the commercial crops, but not to the forage crops. And then when we talk of deficit in the term national deficit has got no meaning, because regional deficits are more important, because fodder is bulky and not economical to transport over long distances. And today when we say changing scenario in agriculture, we know climate is changing. There is no question on that. Increasing population, both livestock and human. When we talk in fodder crops, we have to talk on increasing population of livestock as well. And if we compare the census of 2012 to livestock census with 2019 livestock census, which Ministry of Animal Husbandry carries out every 5 years, and then we can see that there is more than 5% increase in the number of livestock, and more so in the number of milking animals. Milking animals, so the demand of fodder, nutritious fodder is increasing. So increasing demand of quality food and fodder because of urbanization and increasing our purchasing power, people are more and more shifting towards the livestock product, milk product, paneer, ghee, and others. So the demand is increasing. Our resources are decreasing, land holding is decreasing, input is decreasing. Soil water quality is decreasing as well as availability is decreasing. Our genetic diversity is reducing, germplasm base is narrowing down. And most important for forest, that our grasslands, natural pasture lands, rangelands, they are all degrading. So now coming to how forest research network is being maintained in India. First, if we say it was a landmark study by grassland survey done by PM Davadghav and K. Shankar Narayanan. It was published in 1963. It was a great work where they have carried out whole India grassland survey, and ultimately PM Davadghav became the first director of IDFRI, and K. Shankar Narayanan became the director of Kajri, Jodhpur. And then IGFRI Jhansi was established in 1962. AICRP forage crops was established in 1970. And then various other institutions like Kajri, Jodhpur, Noida, Hyderabad, Limited, Ludhiana, they all carried out research on various aspects of forage crops on a limited scale. And then many SAUs like HAU Hisar, PAU Ludhiana, TNU Coimbatore, Anand, Sukasinagar, their forest section or division or agroecology section. So all these forest research network is still working and developing technology for forage. So when we say forest research, it is crucial for livestock protection and livelihood. And I've already said it gained momentum after establishment of IGFRI and Equipon forage crop. Many improved varieties were developed in SAUs as well as an ICR released and recommended for cultivation in different zones as well as specific farming situation. And our focus is on high yield, quality, and specific need of zone and cropping situation. More than 350 varieties and have so far been released or notified in different forage crops. So if I give a brief introduction of our institute, ICRI Jhansi, it was established in 1962, and it is working on all aspects of grassland, forest crops, technology development, dissemination, utilization, as well as policy formulation, and working as a knowledge repository. Headquarters located at Jhansi, and we have 4 regional stations at Avikanagar in Rajasthan, Dharwad in Karnataka, Srinagar in Jammu and Kashmir, and Palampur in Himachal Pradesh. So catering the need of forest research in different agroclimatic zones. Then we have All India Coordinated Research Projects on Forest Crops and Utilization, which is working with 22 coordinated centers across the country, and also 25 voluntary centers also across the country, located in different state agriculture universities, as well as Central Agriculture University, ICR institutes, and also the many NGOs like Urli are also associated with it. So with this network, we are catering the need of forest research, germplasm conservation, as well as all the aspects across the country. All these centers are actively involved in the germplasm collection. So when we say of genetic seed resources, these centers located at different parts of the country are regularly conducting germplasm collection across the country, particularly in their climatic zone, and sharing of germplasm among these stations are being carried out. So when we say of plant genetic resources, naturally a question comes, why? Why? The most important aspect is to develop a variety. So when we talk of a forage crops, what are the improved high yielding variety target traits? What for we are targeting? Because unless and until germplasm are collected, evaluated, and utilized for some target trait, its purpose is not fully justified. So our targets are better yield. Yield means not in the term of seed or not in the term of fruit, but in the term of biomass, green biomass and dry matter biomass. So it is again different from the other crops we say, because here the yield is not seed, yield is not the fruit, but the yield is biomass, green and dry matter. It should be input responsive. It should have high WUE, water use efficiency, and nitrogen use efficiency, and then the nutritional quality. When we grow a fodder, we mainly concentrate on two aspects. It must have high protein and it must have high digestibility, because these two are factors which are crucial for the livestock growth, and less anti-quality factors. Many fodder crops have got some anti-quality factors, and we try to reduce them, like the Bajra, Napier hybrid or Napier grass have oxalates, Lucina has mymosine. In the similar way, these anti-quality factors, when we try to breed, we try to reduce these anti-quality factors. Then tolerance to biotic stress and abiotic stress, we know it is common for all. Then coming to specific cropping sequences and specific intercropping situation and specific niches. These three are very important for us, because farmers are not willing to allocate whole of their land only for the fodder crops. So we have to devise fodder crops. We will have to breed fodder crops. We have to identify the germ plant which can fit into food fodder cropping sequence or fodder cropping sequence, or intercropped with cereals or legumes or the commercial crops. These are the aspects which we especially look for developing a varieties. And then greater persistence and competitiveness. It is very important for the grasses and legumes which have to survive under the grazing situation in the rangeland or pasture land or grassland condition. So if they are not persistent and they are not competitive, they cannot survive under the natural grazing condition. When we say biodiversity in forest grasses and legumes, we say grass belong to wide range of genera. We know over 600 species are currently used for grazing and livestock feed from about total of 10,000 grass species across the world, and over 1,500 species of legumes can be used as forage, although about 60 species developed and widely used across the globe as the forage, in which especially we say the clovers and medics are very, very important, these two groups as the forage crops. When we say our Indian grasslands, I have already mentioned that if we look for forage biodiversity, we have to look in the Indian grasslands, because grasslands are the major source for these such diversity. When we go for collections, when we go for exploration, we also look for diversity mainly in the grasslands or grazing lands. So Indian subcontinent, we know, endowed with rich genetic diversity of forest plant species, which is largely due to diverse ecoclimatic condition and a variety of habitat and niches. And it is further strengthened by tribal and ethnic diversification, plant uses, religious rituals, and also strengthened by influx of genetic material through our past trade routes. When we talk of past trade routes, we know earlier the trade was through the horses, mules, and the animals. So along the trade routes, they have developed the grazing land or the fodder resources. So these trade routes are very genetically enriched diverse material, because the trade routes from all over the country, they actually took the seeds from their grasslands and spread it across their routes, so that their animal, especially the horses, mules, and others, they can get good forage on their route towards the Indian in India or other part. So this is an important aspect. And then in India, we know there is great variation in climate, soil, physiography, and so we have got very heterogeneous nature of grass cover. But if this genetic erosion, there is a threat alarming. Both our land races as well as wild relatives are becoming extinct due to biotic and abiotic interferences, and more so because of lack of knowledge and awareness regarding forages. Lack of knowledge and awareness is very important aspect still, I say, because for most of us, forage is simply grass, legume, or it is weed. So uproot it, remove it, we do not consider its importance. We have worked a lot on the wild relatives of wheat, rice, maize, and now also giving importance on millets, but we have very little information about the wild relatives of our forages. So when we say what should be our strategy, collection and procurement, we need series of exploration from biodiversity zone, center of origin, hotspot area, and we need introduction through correspondence from various international agencies. And these are the agencies which have a rich repository of the forage material like Iluri, ICRISAT, Nordic Gene Bank, USDA, IGER, IBERS in UK. These are the target gene bank from where we mostly procure the germ plant. Then we say some more introduction part. So in the forage crops, some landmarks event where India, I've already mentioned about Gao and Shankar Narayana wrote a book, Grass Cover of India, after nearly eight years of their survey across the country. Then Bohr in 1960 wrote a book, Systematics of Indian Grasses. Professor R. Mishra established a school of ecology at BHU Varanasi. It was a pioneering work on habitat approach to grassland ecology, understanding structure, functioning dynamics of Indian grassland. And then Professor S.C. Pandey opened a school of ecology at Saurashtra University, and especially in the centrist biodiversity, a lot of work has been done. And he was also chairman of the International Rangeland Congress. Third International Rangeland Congress was organized in India in 1978. And then in 23rd International Grassland Congress, IGFRI, both these events were organized by IGFRI. And it was in India in 2015, in which more than 1,200 scientists participated from 80 countries, more than 400 scientists from abroad, from 80 countries participated in this event, and we talked on all the aspects of international, on the grassland and pasture. When we say of grass diversity, we know Indian Gene Center holds rich diversity of native grasses and legumes, which are conserved due to diverse ecoclimatic condition, variety of habitat and niches. I have already talked. And there are a report of 263 genera and 1,291 species of poaceae, of which 21 genera and 139 are endemic. So diversity is found here. And most of our grasses belong to tribe Andropogoni, Panicky, and Aragrust. Especially these Andropogoni tribes are mostly for the arid and semi-arid and arid zone. Pennsylvania mostly for the humid and sub-humid zone. Aragrust mostly we are important for the lower and mid hills. So rich diversity of these grasses are occurring in Western Ghat, Deccan Plateau, Central India, Northwest Himalaya, and Northeast hills. So when we say about 400 species of 60 genera of leguminosae are reported to be useful as forage. These are the legumes, grasses, browse plants, which are for which diversity is found in India. Legumes, if we say Desmodium, lablab, stylo, vigna, macroptylium, centrosema. In grasses, we say Botrocloa, Dicanthum, Cynodon, Panicum, Penicetum, Sanctus, Lazurus. And in browse plants, we say Lucinus, Esbania, Albizia, Bauhinia, Cassia grevia. These are some of the genera where the diversity is found, and they are endemic mostly to India, and they are very important for the forage crops as the forage crops. Then I've already talked about Abadgaon and Shankar Narayanan. They have conducted a lot of survey for nearly 8 years, and they classified mainly 5 types of grasslands in Indian system. Sehma Dicanthum type, Dicanthum sanctus lazurus type, Phragmites saccharum imperator type, Themeda arundina type, and temperate alpine type. These, but now what is left is anybody's guess. We don't know, because after 62, no such survey was carried out, and it is anybody's guess what is left now. Anyway, these are the main part. If we say these are the Dicanthum, Sanctus, Lazurus in the arid and semi-arid part of the country. This is second Sema Dicanthum belt. This is Phragmites saccharum belt extending up to Assam and Bengal, and then temperate alpine cover in this zone, Themeda cover. So these are the major grass cover of India. And so what is required? Need to collect, conserve, and utilize. In case of forages, importance of PGR, plant genetic resources, has so far not got due attention, as perhaps it lacks direct economic value. Please see the direct economic value I have already explained. And more important, human being is a secondary beneficiary, primary being the animal. So primarily our beneficiary is the animal, livestock, and we get it secondary benefit through the livestock product in terms of milk, meat, or other thing, egg and other. So it is we are the secondary beneficiary, unlike other crops where human is the primary beneficiary. There is need for collecting all the available biodiversity in forage crops from different agroclimatic zones, and considering the wide spectrum of land and climate diversity, we are in a unique position to collect plant species growing in such harsh climate. When we say harsh climate, it is again the next point is very important, because forage grass and legumes are ideal candidate for such stress tolerance, because they are growing in the most harsh climate. So naturally they are reservoir of genes for such harsh condition, both biotic as well as abiotic stress. And in future, if we say we have to identify the genes, isolate the genes, they can be easily found in these grasses and legumes. And however, the rangelands are fast disappearing due to rapid urbanization and other development related activities. So what is NAGS? You all know National Active Germ Plant Sites, and IGFRI is the NAGS for the forest crops, we say. Then rangelands, if we say what are the rangeland, many times I have used the term. If you are not aware of the rangeland, it is a slide which gives you an idea what are the rangelands. Rangelands are extensive area unfit for arable farming. Please see, it is unfit for arable farming, where cultivation cannot happen, and are mostly under natural vegetation, where animals graze. So for the rangeland, there are two keywords: unfit for arable farming and where animals graze. So vast terrain and semi-arid tract have the seasonal pattern of animal migration. If we have seen the nomadic and semi-nomadic tribes which are moving across with their livestock herd through the see these plains. But here the quality and quantity of forage availability is reducing. So these can be used through efficient and speedy management. We need their utilization. So what are the regs? Range grasses, we say various forage perennial grasses such as Dicanthum, Saima, Atropogon, Panicum, Botroclo are endemic to India. These grass species have evolved over the centuries and are well adapted to different situations. And a specific genotype of these species can help in increasing the forage production from uncultivable and problematic lands. Our here target is not the cultivable land, but our target is for the uncultivable and problematic land. So besides high yield, we are also looking for tolerance and adaptability and competitiveness of these genotypes so that they can survive under grazing condition, under harsh condition in natural grasslands. So we need to collect and evaluate the diversity available in different parts of the country. Jam plants are growing in very harsh climates, I have already said, such as extreme temperature, moisture, and other abiotic. So they are likely to possess gene for resistance to these abiotic diseases. Chances of presence of these genes in these gases is more as they are growing in non-arable land, as compared to various cultivated crops. And then characterization and documentation is also important from IPR treaty. Then also the soil conservation. We know we have a problem of soil erosion in many parts of the country, and some estimates say that every year we are losing 111 hectare area they are prone. So many grasses have specialized root characters. So they are a good source for encourage resulting in soil binding, and especially in Panicum maximum, Cynodon dactylon, Panicum antidotale, Dicanthum, Chrysopogon, Andropogon. They are the best soil binders. Then also for the reclamation of problem soil. We have many grasses like Guinea grass, berseem, saftal, oats. These are also exhibit tremendous adaptability to different problem soils. Now coming to the research work done at ICFRI, what we have so far done, a brief introduction of the forest research work being carried out in India, especially at ICFRI and AICRP on forest crops. So if we say we have at present more than 10,000 germplasm stored in our mid-term storage. And we have registered 32 novel genetic stocks. We have published 20 catalogs. We have also come out with several descriptors, especially 3. And from our system, we have released 374 varieties from AICRP and 53 from ICFRI by evaluating the germplasm and also utilizing them to develop the varieties. It is a view of ICFRI mid-term storage module, the gate and where we have stored the different kind of germplasm, and for this we are grateful to NBPGR also, which have given us this module in the Indo-US ED program, I think, and also 2 to 3 years back, they have also supplied us with dehumidifier. When our dehumidifier was not working properly. So with this, we have stored more than 10,000 germplasm in this system, which we are regularly rejuvenating also by growing them in a field after a certain period of time. These are the catalogs and descriptors published. We have published a lot of catalogs and also 3 descriptors. We will come to it. So it contains some cultivated crops, and mostly the rangeland crops or the pasture grasses. So these are a bit of fine. And then we have also registered several novel genetic stocks with NBPGR. Like many things, I have given a few photographs. Like this is a pentafoliate berseem. Berseem we know is a trifolium alexandrium, and characteristically it has 3 leaflets. But here we can see there are 5 leaflets, and we have identified and saved the plants which are true breeding with pentafoliate berseem, and it was registered as a novel germplasm if you say pentafoliate berseem pentaval with the name INGR 0904. Similarly, in the bajra, the 4x maintainer lines were maintained. It is basically deployed, but 4S line. And the specific event I will say about the ploidy series in Guinea grass. Guinea grass is mostly occurring in nature as a tetraploid, but we have developed a series of ploidy in it. I will come about in detail later on. But if we say they are all registered, triploid cytotype, tetraploid, pentaploid, octoploid, nonaploid, and now also the 11 ploidy has been so far registered and developed. Now coming to some typical examples where we have utilized the germplasm, utilized the wild relatives, utilized the germplasm from different gene pool to develop varieties, to widen the genetic base. And I will give an example of a few such crops, like one in berseem. Berseem, we know in India it was introduced from Egypt in 1904. And it became the most popular important winter season legume fodder and cultivated in a lot. It has got multi-cut nature, very high quality, very high digestibility, and naturally it became very popular in central and northern part of the country as a fodder. But it lacked problem. It lacked because it has narrow genetic base. It is not native to India. It was introduced from Egypt. So naturally we diseases. And then there was interspecific incompatibility problem. And then we studied and found that the post-fertilization barrier in interspecific hybridization. So what we did? We introduced and collected different species and accessions of trifolium species. So 27 species were introduced through proper channel through NBPGR by studying the gene bank and obtaining import permit from NBPGR. We imported 27 species and 135 genotypes from across the globe. Across the globe, these materials were introduced in India, and we studied it. We studied detailed morphological, biochemical, and molecular markers, and we identified. And then we prepared endogram, and we identified them to find the closure species for hybridization and identification of donors. So the different accessions we say, and a typical pattern we found that all the Alexandrium species, they are clustered together without overlapping. So when there was no overlapping, it means they are each existing the crossing barrier, because all the genotypes of one species were clustering together. There was no overlapping. It was a very peculiar situation we have seen, which is usually not found in other crops, other species even. But in trifolium, it was a very typical situation we observed. And when we observe this, we published this paper in Plant Systematics and Evolution. And then when we saw, then we tried to find out which are the species which have affinity. Then we went out to study the pollen stigma interaction and found where the incompatibility barrier is existing. We identified donors. These are the donors for desirable trait to our target species was Trifolium alexandrinum or Bursi. So these are the donors. Then we developed the regeneration protocol and we developed the embryo rescue techniques because we found that when we were making cross, the cross was not surviving. Embryo was aborting after 4 to 5 to 7 to 8 days in different crops. So we identified the stage where the embryo was aborting. And at that time, we rescued the embryo and we standardized the process, we rescued the embryo, and we started growing the embryo in the tissue culture by developing the regeneration protocol. And we got a huge success, and we published several papers reporting that these are the first global reports in these several crosses, especially in Trifolium alexandrinum into Constantinopolitan, which are published in Plant Cell Reports, Trifolium alexandrinum into Apertum, plant breeding, Lapasium vesiculosum echinatum, we reported in plant breeding in 2018, and plant cell tissue organ culture, we reported alexandrinum with supinatum. With all these methods, we were able to develop the diversity. See, these are the example how we rescued the embryo and we developed the plantlets. In Trifolium alexandrinum, Constantinopolitanum clause, in Alexandrinum into the supinatum clause, and with this we were able to create diversity. You can see now in the F2 generation, a lot of diversity was observed. Similarly, in Trifolium alexandrinum into a pattern, we developed cross with regenerated material, and you can see a lot of diversity has been generated, which served our purpose, because our purpose was to widen the narrow genetic base of Bursi. So a lot of diversity was there, and a peculiar case we observed the seed color variation. It was also a typical case because both the parents have yellow seed, but in the progeny we can see from dark tan to yellow. And it was also a theory to explain how we can explain this. And we explained on the basis of Genia effect, which was published in the journal Genetica in 2019, where we demonstrated how these variations are created and how the genes are playing a role in this. And so our diversity was created. And if I say the success story was manifested in the release of variety. If we say prior to 2015, no Bursim variety was only from 2001 to 2015, only 3 Bursim varieties were released in AICRP system. Only 3 varieties were notified in between 2001 to 2015. But if we say between 2015 to 2022, as many as 10 varieties were notified now. As many as 10 varieties. So it gave a huge success. It gives a huge success, and it is a typical example of utilization of germplasm. How the germplasm was utilized to develop varieties which can be cultivated across the country. Because if we do not utilize the germplasm, if we do not identify its trait and do not utilize it, it has got no meaning sitting in simply the gene bank. So it is one of the example how these can be utilized. And then another example I am giving it of oat. Oat also, it is the most important winter cereal fodder crop, multi-purpose crop used for grain, pasture, forage, and it is now a most important breakfast cereal also. Most of us are consuming oat as a breakfast cereal, and it's a high fat, protein, and mineral content. And when we are consuming it for a breakfast cereal, it is a very good for, especially for lowering the cholesterol and also maintaining the sugar balance. So for the diabetic and for the persons with high cholesterol, what is a very good. So how we utilize viral germ plant or the relative in Avena sativa? It is a case. One was Avena sterilis. Avena sterilis is another member of primary gene pool, and we know Avena sativa, like the wheat, is 2n is equal to 6x is equal to 42. And we crossed it with Avena sterilis, different genotypes of sterilis after evolution were used, and they were crossed, and we developed fertile hybrids. And then 2 cycles of back crossing was done, and it resulted in wide variability, from which we selected suitable varieties for different locations. So it was another example where Avena sterilis primary gene pool material was used. Second is how to use the secondary gene. Avena americana, we targeted Avena americana. It has very high protein. Actually, why we targeted Americana? It is high protein but very low biomass. Whereas Avena sativa has low protein but high biomass. And then it was an hexaploid, it was a tetraploid. So chromosome number was not matching. So when we crossed, we got the F1 hybrid, which was sterile. So when it was sterile, we started 2 ways. One was the back crossing, and after back crossing, we could get stable hexaploid, and these stable hexaploid had many desired traits of Americana. And the other aspect we went on, we colchiploid to raise the level to the decaploid. And when we sell, we got an array of cytogenetically stable lines of different types. These stable lines again became a material for our future breeding program. So if we say polysomic stocks, you can see 2n is equal to 42, 44, 44, 46, 50, 64, 68, 70. All these were created in this manner. So in this way, we utilized it. We developed one particularly variety also. By this way, you can see leaf, it has very high protein as compared to the other cultivars. Nearly, if we say this, normally oat lines have 8 to 9% crude protein. It has 11 to 12% crude protein. And also the dry matter digestibility in oat, it is normally nearly 50%, it is 57%. So in this way, we developed cultivar with high yield and quality. And it was also released for cultivation in almost all part of the country, northeast, northwest, south hill zone. Then after giving you successful example in 2 cultivated crops, I'm again talking of some range grasses. The range grasses or the pasture grasses, which are grazed and which are not cultivated. It is one example of heteropogon contortus, which is occurring mainly in the central part of the country and lower hills. So we collected 235 germ plants from 10 states. And a total of 39 traits, including 14 metric traits, 14 non-metric traits, and 9 nutritive parameters were evaluated. And a germ plant catalog was published with ISBN number. And then the same thing was also published in the International Journal of Tropical Grass and Forages Tropicalis in in 2021, where we also developed a core germ plant set, a core germ plant set with almost more than 98% diversity conserved. We have developed it. Another example is the marvel grass or Dichanthium botryopoda complex. Again, we developed we collected 498 germ plants from 14 states. We evaluated it for 27 metric morphological traits, 18 non-metric traits, and 9 nutritive traits. And we published the catalog and also developed a core collection of this. And then based on the core collection, we also published the paper in the Journal of Agricultural Sciences in 2022, only in the previous year. It was published in Journal of Agricultural Sciences, a course set. Then in Guinea grass, earlier it was called as Panicum maximum. Now it is called as Megathyrosus maximus. Here 152 germ plants, including 27 procured from Africa and 1 from Brazil, and 124 indigenous collection were evaluated for 21 metric traits, 13 non-metric traits, and 9 nutritional parameters. It was also published catalog with ISBN number, and then it was also, we also developed a core germ plant set, you can say core germ plant set. It was published in Plant Genetic Resources, Characterization and Utilization in 2020. So it is another. Now another important grass, Sehima nervosum. We collected 81 genotypes from 8 states. States evaluated it for 16 numeric traits, 8 non-numeric, and 9 nutritive traits. And we published the paper in Crop and Pasture Sciences, a CSI Euro publication in 2022. To again, it was published. So in this way, we are talking about the range grasses where we have successfully utilized them. And then this is another example how we utilized the various germ plants to develop world's largest ploidy series in Guinea grass, and it is also entered in Limca Book of Record 2018. And you can see 3x genotype, 4x genotype, 5x genotype, 6x, 7x, 8x, 9x, 11x. If we say in the nature, we get mostly 4x.
Hello, sir. Your voice is not coming, sir. Roy, sir.
डॉक्टर रॉय, आपकी आवाज नहीं आ रही है।
रॉय सर, आपका आवाज नहीं आ रहा है।
सर आपकी साइड से फ्रीज हो गया है।
हाँ, यस। हाँ। मंडल जी,
सर उनका सिस्टम शायद रिस्टार्ट हुआ है।
हाँ, ओके
कनेक्ट
ओके।
हो गए हैं।
हाँ,
उनको एक बार फोन कर लीजिए।
यस। हाँ, चाइनीज थोड़ा नेटवर्क का इश्यू है, हम थोड़ा सार से
सर आप लोगों को आवाज आ रही है? हेलो सर, डॉक्टर रवि।
अरे मंडल जी सार आ रहे हैं मगर
आवाज आ रही है सर आपको?
हाँ नहीं आ रहे, कम आ रहे।
कम आ रही है तो तेज कर देंगे, ठीक है।
ना ना
अभी ज्वाइन नहीं किया सर ने।
हेलो हेलो मंडल जी।
यस मैम।
हाँ ये लाइक मैं यहाँ पे स्पीकर लगाई हूँ,
हम्म,
तो अगर मतलब ऑडियो ऑन करके छोड़ देती हूँ, मतलब मैं अपने वाला, तो स्पीकर से आवाज जाएगी तो तेज जाएगी। वो करके देखूं?
करके देखो।
हाँ वो
करके
करके
देखो।
देखती हूँ एक बार जो है।
हेलो हाँ मैम,
हाँ
सार आ
हाँ
रहे हैं मैम, दोबारा ज्वाइन कर रहे हैं।
ओके सर आ रहे
हाँ
हैं, ठीक है ठीक है,
हाँ,
तो
वो
सर कर
एक्चुअली
दे ज्वाइन
ऑलमोस्ट
कराइए।
यस लेक्चर खत्म
ठीक
होने
है,
वाला है।
उसके बाद कर लेंगे, ठीक है।
हाँ सर दोबारा आ रहे हैं, मंडल जी एक बार देख लीजिए।
हाँ ठीक है, अभी ज्वाइन नहीं किया उन्होंने।
ज्वाइन नहीं किया ना सर ने?
नहीं अभी नहीं किया।
हाँ मंडल जी सार नहीं आए ना?
नहीं सर नहीं आया अभी,
अच्छा, ट्राई तो एक्चुअली वो कर रहे हैं, बोल रहे हैं।
क्योंकि यहाँ का नेटवर्क इशू लग रहा है।
हाँ नेटवर्क इश्यू, वो सार बोल रहे हैं ऐसा हमारा पास नहीं है नेटवर्क इश्यू ऐसे बोल रहे हैं, बट फिर भी कभी कभी हो सकता है, बीच में इंटरप्शन होता है। जस्ट उनका ऑलमोस्ट लेक्चर खत्म हो गया, बट थोड़ा जस्ट लास्ट में है। दोबारा एक बार कॉल करूंगा मैं।
हाँ दोबारा कर लीजिए एक बार।
हेलो मंडल जी, हेलो मंडल जी, एंड मैम ज्योति मैम,
ठीक है हाँ
sir
रवि
actually ma'am there is a network issue from SAR side
ओके सो
from their
ज्वाइन
institute.
नहीं कर पा रहे हैं सर
ज्वाइन नहीं कर पा रहे हैं और मैम
हाँ
एक्चुअली आई थिंक एक दो स्लाइड ऑलमोस्ट ही इज कंक्लूडिंग ही टोल्ड.
ओके ओके तो ये चेक करूँ जो 1 मिनट
हाँ फिर मैम एक बार मैम थैंक्स बोलकर फिर मैं ये कर दूंगा.
ठीक है हाँ कर लीजिए
And now it is that actually unfortunately SAR slides are not that means there is a network issue from their end and the slides are not coming and there is almost the topic is over. And now it is the time to thanks to Dr. A.K. Roy for a detailed and highly informative lecture presentation on forage genetic resources and how it will be useful for livelihood and cattle. And he mentioned grass diversity, rangelands, and what are the achievements and technologies of IGFRA. Also, he covered almost in the detailed way. And really, sir, I am very thankful to you, sir. And sorry, we are not able to connect you. That is it. Thank you, sir. And dear participants, give me time for 5 minutes. We are checking for next participant. Yes. Next speaker.
12:15 so 10
12:15.
minutes
10 minutes.
break
10 minutes, 12:15, and please be in online. Thank you. मंडल जी एक बार हम वीडियो चेक कर रहे हैं.
ठीक है
जो नेक्स्ट स्पीकर का थोड़ा
कीजिए चेक करो सर.
हाँ यस यस प्लीज. मैम प्ले करिए.
हाँ अब जा रही है आवाज
नो नो आवाज नहीं आ रही मैम.
अभी भी नहीं जा रही है
हाँ नहीं आ रहा है आवाज. हाँ
नहीं मैं स्पीकर मोड में अगर इनकी आवाज यहाँ पे आ रही है तो वो आवाज स्पीकर के थ्रू जाएगी ना.
आना चाहिए मैम बट
देख
अंकुर
जा रही है आवाज
नहीं मैम आवाज नहीं आ रही मैम.
कुछ आवाज नहीं जा रही है
हाँ कुछ नहीं जा रहा है एक्चुअली उनका प्रोग्राम चल रहा है समथिंग कुछ सॉफ्टवेयर ये होगा मैम इसका ना.
अंकुर क्योंकि मैं स्पीकर तो ऑन करके रखी और तब भी नहीं जा रहा है मतलब मेरा अनम्यूट करके.
अच्छा
तो मैं सोची उसके थ्रू जाएगा बट आप कह रहे हैं कि कुछ नहीं आवाज जा रही है.
हाँ मैम आवाज नहीं आ रहा है. प्रोग्राम तो चल रहा है मैम ऐसा ना प्री रिकॉर्डेड प्रेजेंटेशन है ना तो बट आवाज तो नहीं आ रहा है.
यहाँ तो आ रहा है हमें.
कहाँ आ रहा है
मतलब मैं सुन पा रहा हूँ मैडम की आवाज को.
ओके अच्छा आप सुन पा रहे हैं फिर
जी जी
रवि
बिल्कुल.
आपके यहाँ तो कुछ नहीं है. नहीं मेरी आवाज नहीं प्रेजेंटेशन की आवाज मैं कह रही हूँ. प्रेजेंटेशन में
हाँ
अभी चेक कर रही थी वो
नहीं
आवाज
मैम
जा रही
नहीं आ
थी.
रहे प्रेजेंटेशन की
हाँ
आवाज नहीं
प्रेजेंटेशन
आ
का
रही.
यस यस प्रेजेंटेशन का आवाज. हाँ यस
स्लाइड में कुछ ऑप्शन होगा स्लाइड के अंदर वो
इधर मैम कुछ दिखा रहा है ये वाला मैम ये करिए. ना ना इधर क्लिक करने से नहीं आ रहा है मैम. जो फोटो के पास है ना प्ले. हाँ ये तो प्रोग्राम चल रहा है बस है आवाज. मैम इधर स्पीकर भी देखिए इधर स्पीकर ऑप्शन है ना फोटो के ऊपर.
फोटो के ऊपर
हाँ स्पीकर भी दिख रहे हैं. हाँ हाँ यस, उसका तो हाई कर दिया ना आपने।
हाँ, आ रही है, वो भी नहीं आ रही है।
नहीं आ रहे मैम, और कुछ होगा सेटिंग्स।
अ, मंडल जी ने चलाया तो भी नहीं आ रही थी।
ना ना, उसका भी नहीं।
ये, आवाज जैसे स्पीकर, लाइक मैं बोल रही तो मेरा जा रहा है ना,
हाँ आपका तो आ रहा है मैम। हाँ
जैसे ये बोल रहे हैं तो इनका आवाज कैच करके नहीं ले रहा है स्पीकर।
इसका वीडियो प्रेजेंटेशन का कुछ मोड।
मैडम मैं ट्राई कर रहा हूँ अपनी साइड से।
हाँ मैम, एक मिनट
ठीक है,
रुकिए
मैं
मैम।
स्टॉप कर रही हूँ, स्टॉप कर
हाँ
रही
मंडल जी ट्राई कर रहे हैं मैम, एक मिनट।
हूँ। ठीक है।
Hello, my name is Martin Zonneveld. I'm the team lead manager of the World Vet Network, responsible
मैं तो सुन रही हूँ मंडल जी।
collection of
आवाज आ रही है?
65
हाँ मैं सुन रही हूँ,
आवाज आ रही है, आवाज आ रही
आ
है। हाँ
रही है।
the largest in
ठीक है मंडल जी, आप करिएगा वहां से फिर प्ले, जो आ रही है।
अब दूसरी स्लाइड है, बताना कि आवाज साफ है या क्या है?
मुझे तो साफ साफ सुनाई दे रही है।
the nation of Turkey.
औरों को भी सुनाई देना चाहिए।
So here's the outline. We start with an introduction and
सुनाई
we
दे
end
रहा है?
with some
हाँ यस, हाँ अच्छा आ रहा है मंडल जी। हाँ
go through the presentation, through the different steps
हाँ
of
यस।
regeneration.
देख लो थोड़ा कहीं ऐसा हो यूजर
थोड़ा ना ना, थोड़ा साउंड बढ़ाइए आप थोड़ा।
बढ़ा
हाँ
दिया, अब दोबारा
Oh.
ट्राई कर रहे थे।
हाँ हाँ।
दोबारा बोलना, आवाज कैसा आ रहा है?
नो अभी तो कुछ नहीं आ रहा है, रुक गया वो।
ये अभी
So here's an outline. We start with an introduction and
आ
we
रही है, थोड़ा
हाँ
बढ़
यस
गया
यस, हाँ अच्छा है, अच्छा है, क्लैरिटी है।
go
हाँ
through the
परफेक्ट है मंडल जी,
different
परफेक्ट।
steps of the regeneration of
ठीक है।
ओके ओके ओके।
germplasm of
हाँ
काम चल
vegetable
जाएगा?
crops.
हाँ हो जाएगा
हाँ हाँ
मंडल
हाँ,
जी।
बहुत अच्छा है।
So let's first have a look at the concept of regeneration.
12:15
आ रही
में शुरू करेंगे।
A seed
आवाज आ रही है?
आवाज आ रही
the
है। हाँ
renewal of germplasm accessions by sowing seeds or
यस यस।
planting vegetative
ओके मैम, ओके।
material
मंडल जी थैंक यू।
and
हाँ
harvesting
हाँ।
seeds or plant materials which will possess the same character.
हाँ थोड़ा ब्रीफ, मैं इंट्रोडक्शन बोल दूंगा, फिर इसके बाद आप प्ले करिए।
So this is
ठीक है मैम, 12:15 ना मैम क्लास।
15 5 मिनट के बाद कर लीजिए। 10 हुई है।
the same. So let's first have a look at the concept of regeneration. So regeneration, we sometimes also say seed multiplication, of germplasm accessions by vegetative materials and harvesting the seeds or plant materials which will persist
आवाज आ रहा है अच्छे से आ रहा है
as the original population. So this is important. So let's go further. So
ठीक है मंडल जी हेलो ज्योति मैम
हाँ रवि
स्टार्ट कर लो मैम
हाँ हाँ आप कर लीजिए
welcome back dear participants now we have a current speaker actually that is he sent unfortunately he is not able to attend the presentation then he sent one pre-recorded presentation we will play that one but firstly let me introduce our speaker he is Dr. Martin Johnnyberg he is a gene bank manager World Vegetable Center Taiwan and he started his career as an associate scientist in forest genetic resources biodiversity and afterwards he joined as a scientist in PGR and then again he is a gene bank manager that means responsible for the world largest international vegetable gene bank Taiwan and he is a head of gene bank and he is that means the national gene bank the gene bank is around more than 65 accessions of vegetable crop resources and he involved actively in spatial analysis of plant diversity and distribution and main he concentrated on the traditional African vegetables that means conservation he particularly concentrated on that one and he worked and he is a conservationist and he involved in the capsicum diversity in Andean region of diversity and he undergoes the research of screening of Vigna wild relatives for abiotic and biotic stress tolerance and he is having a lot of awards and recognitions even though I given a very small event and he is a member for fundraising for public research and development he involved in nearly 12 million US dollars that means fundraising for 18 projects and he is a lead proposal writer of $9 million US dollar and he is having a lot of publications are there around 41 research papers in international reputed publication and other extension bulletins are there and YouTube videos and lot of videos also you will get in YouTube and is vast contributions in conservation of vegetable genetic resources. Now we are playing the presentation of a pre-recorded presentation Please, I am requesting Mandalji.
Sir.
Recorded presentation pre-recorded ना present करिए. If you have any doubts, please kindly post in chat box.
I'm the Gene Bank Manager of the World Vegetable Center, responsible for our vegetable germplasm collection of around 65,000 accessions. This makes it the largest international public gene bank for vegetable genetic resources. The topic of this lecture is about good practices in germplasm regeneration of vegetables crops, so that we can have sufficient seeds of our gene bank accessions of the vegetable crops that we conserve. So here's an outline. We start with an introduction and we end with some conclusions, and we go through the presentation through the different steps of the regeneration of the germplasm of vegetable crops. So let's first have a look at the concept of regeneration. So regeneration, we sometimes also say seed multiplication, is the renewal of germplasm accessions by sowing seeds of planting vegetative materials and harvesting the seeds or plant materials which will possess the same characteristics as the original population. So this is important because we want to keep as much as possible the same genetics, the same characteristics of the varieties that we have collected in the field or from the populations of crop route relatives that we have collected in protected areas or outside. So why do we want to regenerate? First, to increase the quantity of initial seeds or planting materials, because when we go to the field for germplasm collecting, many times we only have a limited amount of seeds because of logistics or simply because there are not sufficient seed of a variety on farm or in the wild. Then secondly, to replenish seed stocks or plant materials in active and base collections with low seed viability or low quantity of seed, because over time, seed viability will go down. So sooner or later, whether it's after 30 years, 40 years, 50 years or longer, we need to regenerate the accession again to have again fresh seeds. In some cases, we also have low quantity of seed because we want our gene banks to be used. So we distribute seed to researchers, breeders, others. So when we are out of seeds, in this case, our active collection, we should regenerate seed again. So this also goes to the next point, to increase seed of requested accessions that cannot be distributed because no available inventory. So this definition comes from this link that you can see here, cropgenebank.sgrp.cgir.org. This is a website which is currently not active, so it's not maintained by any organization, but this was the website of a program which was called a system, actually I forgot the name, but it was part of a genetic resources program of the CGIAR, and a lot of the material that is on this website is still valid for plant genetic resources management currently. So let's go further. So the aim of regeneration is to keep the multiplied seed of an accession as close as possible to the original seed samples. So you could say that this is actually opposite from breeding, from the objectives of breeding, where we want to select for improved material. We want to look for material that has specific traits, and we want to develop material by crossing different lines. We want to create new material. In our case, regeneration of gene bank accessions, we want to keep the existing materials so we want to keep as close as possible to original seed samples during our regeneration. And that is often a challenge because when we grow accessions out in the field, there might be selection pressure because of specific weather conditions. For example, if there's a lot of heat, and then maybe you, with the plants that you use to do the regeneration, you automatically will do a kind of selection towards those plants that are more tolerant to the heat, to give you an example. Because we only use a small sample of seeds from our seed sample to regenerate, we will always create genetic bottlenecks, larger or smaller. So with each regeneration cycle, we will lose genetic diversity. And that also drives us more further away from the original seed samples, and we create also genetic erosion within our seed sample. So we identified 4 principles. We call them the MAMA principles, and I consider these like the basics to use the basic guiding lines when we do regeneration. So think about MAMA when you do the regeneration. It consists of the following 4 points. First, minimize selection pressure, attention to all accessions, minimize loss in genetic diversity, and 4th, avoid crossing between accessions. So we will go in more detail about these 4 points of MAMA in the following slides. The first principle is minimize selection pressure. So we want to randomly select seed from our initial seed sample, which could be the original seed source from the field, or this could be your seed lot in your collection. So randomly select seeds for regeneration. Of course, your seed needs to be healthy. So you can discard seed that are not healthy, but otherwise please randomly select. Then ideally grow accessions in a similar environment as the original collection site. So by mimicking this environment, you will also reduce the selection pressure. Instead, if you grow the accession in a completely different environment, then you might start to include some environmental pressures where perhaps only a few plants of your accession will thrive, and with that you start to include selection in your regeneration. So again, try to grow your accessions in a similar environment as the original collection site. This can be, you can do that very sophisticated by checking the climate data in original collection sites, and then look for a similar environment in where you can regenerate, or you can do that with some rules of thumb. For example, if you know that accessions are being collected in the lowlands, okay, you make sure that also your regeneration is being done in the lowlands for those accessions. If other accessions are being or were collected in the highlands, okay, then do your regeneration also in the highlands of those accessions. Just think about it and check the original collection sites before you grow them out. And then produce and harvest seed from various plants. If you just harvest seed from one plant, not only will you reduce the genetic diversity in your sample, but you will also only include the specific traits of that plant, which might be slightly different from other plants in an accession, because within an accession we have genetic variation. We want to keep that genetic variation and not towards specific traits of one plant. The second principle of MAMA is to take attention to all varieties. So it's important that you take care of both well and poor performing accessions. It might be that some accessions are not well adapted to your regeneration site. Still, you take care of them because they're part of our genetic resources pool. While they may not perform well at your specific site, they still may maintain traits that are of interest for breeders and researchers. The third principle is to minimize loss in genetic diversity. So that's important because in the end, what we want to do is to conserve genetic diversity. However, by regeneration, we will create bottlenecks. So our genetic diversity will always decrease after we have collected the seed from the field. Because we will generate bottlenecks by each time when we do a regeneration, only select a subsample of the seeds of our original seed sample or the seedlet that has been regenerated before. So we need to minimize the number of events of regeneration because they will increase the number of events of genetic bottlenecks. And per regeneration, we need to use sufficient plants per accession. And this is a balance between what is recommended in theory and what you can do in practice. Because maybe in theoretically papers recommend you to grow 100 plants, but maybe you only have space for 20 plants per accession. Okay, then you still grow those 20 plants per accession. The important point is that you know that you will create a genetic bottleneck and it will even more important to minimize the number of events of regeneration. So that means that you need to produce sufficient seed in one regeneration cycle so that you don't have to do any further regeneration. You need to produce seed of good quality and produce seed of good health. So regenerate an accession once and do it good. And the final principle of MAMA is avoiding crossing between accessions, because we don't want to develop new materials by crossing two lines or two accessions. We want to keep the genetic integrity of our accessions. We want to have our accessions to be as close as possible to our original seed samples. So I crops that outcross or have so concept.
फोन किया था कल बोल रहा था ना फिर बोल अभी फोन किया था मैम म्यूट करिए
मंडल जी
हाँ
यस कुछ दिक्कत आया वहाँ
ये रुक गया क्योंकि वो
हाँ
ये के सिंह हुए जो बात में तुम मान ले बोल दे हाँ सेवक क्या है बोल के छोड़ दे
जो बोल रहे हैं उन्हें बाहर कर दो क्या उनकी वजह से डिस्टरबेंस हो रही है
यस सर हाँ यस यस मंडल जी
चलते मीटिंग रुक गई
Let's go to the preparation. So in order to do a proper regeneration and to follow the 4 principles of MAMA, we need to know the reproductive biology, especially because we want to avoid crossing between accessions. So identify the reproductive biology is crucial, and there's a lot of literature about it. So please investigate, do literature research, consult your peers about what the pollination type is, what the pollination mechanism is, and what kind of methods you can use to avoid crossing between accessions. So here you see 5 examples from the database from WorldVeg, some which is basically based on literature. for defining the pollination type or pollination mechanism, and then the method used, following literature, but also our own experience. So you can see that for isolation, sometimes you need to isolate. If you just grow accessions in the open, which means that insects can come to cross-pollinate, or you have wind pollination, you need to isolate accessions at very large distances, so that's often not possible. It means we need to isolate them, and we will come to that in a moment. So please take into account that wild relatives of crops can have different reproductive biology compared to domestic crops. For instance, while the cultivated tomato, or better said, the domesticated tomato, is known to be selfing, several wild relatives, or maybe all wild relatives, are outcrossing or have a mixed pollination system. So they require different methods of regeneration. Check the literature, and flower morphology can provide some hints. So, for instance, the length of the pistil and anthers can give you some indications whether this specific species, whether it's a wild relative or a domestic crop, is selfing, where the pollen will fall through gravity on the anther, on the pistil, or whether it requires some insects to do cross-pollination. So documentation is also crucial, and poor documentation is often a cause for mixing accessions up in the field, so that you have accessions that don't correspond anymore to the original seed samples. So remember, our objective is that the regenerated material is as close as possible to our original seed samples. So make a list of the selected germplasm. This is still on paper, but maybe you also want to use technical devices. I still prefer paper because it's easy to go to the field. It's easy to make notes, but you check yourself what's most convenient. Also label the seed of the accessions to be regenerated. Label in the nursery, label in the growing, label as much as you can. So labeling is really a vulnerable aspect because of the different steps in the regeneration. So from your fridges or freezers where you have the seed stored, to the nursery, from the nursery to the field, from the field back again for seed harvesting, and then again to the drying and to the seed storage. So label these accessions through all processes, and label all accessions individually with long-lasting labels and with permanent ink. Keep field maps to monitor accessions through the crop season. Labeling, labeling, labeling. And then seed treatments are important also in your preparation. So know what kind of seed treatments you should do to let your seed germinate. And this is specifically relevant if you do nursery, but also if you do direct sowing. This helps you to prevent infection of diseases in the seed and seedling phase. For example, by coating seed with fungicide. It's in the case of chemical treatment, it helps you to remove virus and virus particles from the seed surface, which is now a specific problem in Solanaceae crops, but also in other crop families. Some seed treatments are important for seed dormancy breaking. This is especially relevant in the case of crop-out relatives. So dormancy can be broken by heat treatments or by chemical treatments. These are crop specific, so it requires literature or research. And then seed priming, which is basically to enhance germination, germination speed, overall germination, and germination uniformity. So good practices in seed germination: investigate the seed biology of target species. Remember that seed priming are pre-sown treatments that enhance germination. So overall germination, germination speed, and germination uniformity. And then seed dormancy blocks completion of germination of viable seeds. So please check if your crop requires a seed dormancy breaking protocol. So do not assume that non-germinating seeds are non-viable seeds. It may be that dormancy has not been broken yet. So you always need to save seed of your original sample for 2 or more attempts of germination. And you can do TTC tests to check if the seed is really viable or not. So again, do not use all your seed during the first nursery. I'm emphasizing this because sometimes when we work with breeders, they got so enthusiastic that they just use all seed in the first time, and they find out that the seed is not germinating because there's a seed dormancy issue, but then there's no seed left for a second attempt. Well, for breeding, this is okay because they will just focus on other lines. For gene banks, this is fatal because we have lost an accession. So let's now go to the nursery stage, which is important in vegetables because most vegetables are being transplanted in the field. So what are the advantages of direct sowing versus nursery? So direct sowing often is being done in field crops, in crops that have high germination, and direct sowing also helps you to have a high number of plants per accession so that you can minimize genetic diversity loss. Example of crops in our gene bank where we do direct sowing is mung bean and soybean. However, for most crops, in our gene bank, we use nursery because they have either low germination and dormancy issues, so they need specific treatments and specific care. Many of our crops have intensive management, and examples are Solanaceae and cucurbits. Relevant for most vegetable crops, the disadvantage is that you have a limited number of seedlings per accession because it's not feasible to have a very high number of plants per accession in your nursery, at least not in our gene bank. In our gene bank, we can have up to 20, 25 seedlings per accession, so that we can grow out, transplant in the field about 20. So this is a risk of genetic erosion, but the benefits in our case are much larger. And in many cases, it's the only way to properly grow and regenerate horticultural crops. So nursery is relevant for most vegetable crops. So for your nursery, a good medium is important. I mean, I'm saying important a lot of times in this presentation, but yes, in all steps, let me put it another way, there's so many steps and you need to take well care of each of the steps. So your nursery, or the medium for your nursery, needs to be free from soil-borne diseases such as fungi and bacteria, free from weed seed, be permeable, so good drainage, good on retention capacity, and there should be enough fertilizer. The next stage is transplanting. So in transplanting, we transport seedlings from our nursery to the growing fields. So ideally your nursery should be close to your growing field. Be careful because young and weak seedlings can be damaged and eventually die, either before transplanting or during transplanting. And seedlings need to be hardened before transplanting, for example, by reducing watering. Again, labeling is important. So during transplanting, there's a high risk of mistakes in labeling because Yeah, it might be complicated in the field. People make mistakes, so checklists to prevent mistakes. Labels in the field must be settled down in advance. Check again to ensure materials are planted in the right plot. So if you're responsible for regeneration, be there in the field. Double check of accession number or variety should be done before movement. And train and oversee field staff who are doing the transplanting. And then after you transplanted, immediate watering can help the plant to recover and stand erectly quickly. So suitable moisture supply is the first and most important component for the plant growing fine. And here you see a few pictures of how we do it in Taiwan. The next step is field management. So, of course, each crop is different and requires a different agronomy, but there are a few principles that apply in general. So remember about the reproductive biology, why it's important to know it, because we want to avoid crossing between accessions. So to do that, we can use net cages, tunnels, and sacs to control insect pollination and pest incidents, and to prevent insect pollination, or to stimulate pollination. So we want to prevent insect pollination between accessions, but we want to stimulate pollination within the plants of an accession. So we want to prevent insect pollination between accessions. That's one of our NEMA principles. But we want to stimulate pollination within an accession to minimize the loss of genetic diversity. So those are two different important concepts. So net cages for multiple accessions can be built to prevent insect pollination. So you make sure that within the net cage there are no insects. So that also requires really intensive pest management and control within. So once you know there are no more insects in the net cage, it's relatively easy to control. And then you can grow multiple accessions within the net cage. And then you can either do hand pollination, or these crops, they do some selfing, and you can produce sufficient seed per accession. Or you can use net cages and sacs for individual accessions. So here you see a few pictures where we have net cages, in this case for brassica, where one accession of brassica is grown per net cage. So you see several plants of a brassica accession within a net cage. And you can also see a sac for onion, where different plants of the same accession are closed in one sac. And within those individual net cages, or net cages for individual accessions, there will be pollinators introduced, bees, flies, or whatever, that then can do a lot of regeneration within those bags. And then we have the net bags. This can be cheaper compared to net cages, and is relevant for seed mating and selfing of insect pollinated crops, and also relevant for selfing of wind pollinated crops. So what you often do in the case of insect pollinated crops, such as minicocurbits, you look for the flowers, the female flowers that are almost grooming. So you close them so that you prevent pollination, uncontrolled pollination, and you collect the pollen from male flowers of the same accession on other plants of the same accession, and then you cross-pollinate by hand the flower that you have identified, and then you close the bag again. So that is hand pollination, and that's a way of controlling, so that you make sure that you use pollen from the same accession to fertilize the female flowers. Again, to minimize genetic diversity, you should use male flowers from other plants of the same accession to put pollen in those female flowers. Check that close packs may have negative effects on the microclimate for flower and fruit development, so that requires some care. And then staking. So in many vegetables, they require also staking, such as tomato, but also eggplant. For instance, we should avoid that they fall down because they get top heavy because of the fruits. Some are just climbers. So it's staking supports the soft main stem to grow up. Let the plants grow not too wide. It's easier for management. Support the plant for not lodging when there's heavy fruiting. So this is especially relevant for tomato, pot legumes, and cucurbit. Let's now go to the seed harvesting and extraction. So remember to harvest seed at physiological maturity, which is different from commercial maturity in some crops. For instance, cucumbers, they are being sold when they are green, immature, but we need to harvest the seed when they are mature. Yellow, often, or brown. The same with eggplant and with other crops. So please check when it's the physiological maturity. And then labeling remains important. Whether you have big fruits, then you write a label maybe within the fruits like cucurbits. Or when they are in sacks, you have your label inside the sack, like in this case for these legume pots. So in seed extraction, we distinguish wet seed versus dry seed extraction. So wet seeds include cucurbit solanaceae. So we need to make sure that there is a plastic label where there's the code written in a permanent pen. So the ink is not washed away. And then when we extract dry seeds, such as in the case of crucifers, brassicas, legumes, and amaranthus, you still have to have a label to make sure that you know at each time what the identity is of the seed that you are extracting. So here's an example of pictures of wet seed extraction of cucurbits. And here for pepper. So we use an extracting machine, and then through the whole process of watering, drainage, watering, we get the seeds and we keep the label all the. And this is an example of dry seed extraction. The seeds are being threshed, in this case of legume pods. And then post-harvest seed treatment. So wet seed extracted seed, normally seed moisture content, according to our experience, is between 30 to 50%, while of dry extracted seed, it's between 80 to 25%. So normally we let the seed dry for a few days under the shade, pre-drying up to 12 to 15%, although for some crops we have other treatments, for instance, tomato. It requires also some fermentation. So again, crops, there might be exceptions for specific crops. And then after that pre-drying, we dry them in our drying chambers at maximum 20 degrees Celsius and maximum 50% relative humidity until we have a seed moisture content of about 4 to 7% for seed storage. This is crucial. Without good drying, there's no good seed quality. And with no good seed quality, we will probably have to regenerate another time, and this will recreate, again, more genetic bottlenecks, and we will lose more genetic diversity. So very important. And again, label, 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 also at this stage. And then finally, the seed packing before it goes again. Before the seed regenerated and harvested goes into a seed storage. In this case, we use aluminum foils, which is following international GeneBank standards. And again, labeling, labeling, labeling. So the conclusions. The greatest, so regeneration is complex, requires many considerations, and in order to minimize genetic diversity loss, you probably have to plant a lot of, you have to grow out a lot of plants per accession. However, the greatest risk of doing proper regeneration is wanting to do perfect regeneration. So even if you don't have all the means to do perfect regeneration, don't wait, just start with your regeneration, because otherwise you will create a regeneration backlog and the seed will be eventually die. So just start and do your best. It's an art regeneration on the base of scientific knowledge, experience, and available resources. And regeneration protocols differ among crops and between crops and their wild relatives. So that makes regeneration, and especially for vegetable germplasm, to be knowledge intensive. And why do I say especially for vegetable germplasm? Because it requires also many horticultural skills as well. Remember the 4 MAMA principles: minimize selection pressure, attention to all accessions, minimize loss in genetic diversity, and avoid crossing between accessions. And identify reproductive biology to develop the appropriate regeneration protocol, especially to avoid crossing between accessions. Develop your appropriate pre-sown seed treatment protocol so that you can have better germination by seed priming, but also to break dormancy in case that's necessary, and also to have clean seeds free of pathogens. Determine your crop management protocols, and again, ensure labeling in all stages. So I wish you all the best with your regeneration activities, and if you have questions, please write to me. We will stay in touch. Thank you again for listening, and good luck with the rest of the course.
Thank you. Thank you, Mandalji, for playing the pre-recorded presentation. And actually, he is in a business tour. That's why he shared the pre-recorded presentation. And now, thanks to our speaker. Sir, thank you very much for your valuable presentation. And it is highly informative on vegetable genetic resources regarding processing, harvesting, or any precautionary measures. And sir, thank you very much. And dear participants, if you have any doubts regarding the vegetable genetic resources, you can post in our chat box, or you can mail to our PGR training. Then we can interact with him. Then he will give answers and we will share it with you. And meanwhile, we will send presentation also. His presentation we will forward you. And thank you very much. And we can assemble at 2:30 p.m. We have another practical session is there in afternoon session. That is seed viability assessment by Dr. Sherry Jacob and Dr. G.D. Harish. Let us 2:30 we will join. Thank you.
Thank you, sir.
Thank you, Vijay Mandalji. Practical is a seed viability assessment by Dr. Sherry Jacob and G.D. Harish. And let me introduce our speakers. And Madam is Dr. Sherry Rachel Jacob. She is a senior scientist, seed technology, Division of Plant Conservation, Germplasm Conservation, NBPGR. And she is the curator of cereal crops, that is wheat, barley, and maize at National Gene Bank. And she is working as a rationalization and documentation of trait-specific information of wheat, barley, and maize. And she is working on seed viability test and monitoring all the habits of seed processing, testing, and everything. Total management activities of curator of the cereal crops. And she is an outstanding student, as she got a recipient of IRA Merit Medal, and she is a Young Scientist Award also. And she registered 17 genetic stocks registered with the NBPGR, INGR in wheat. And beside all these things, she is a best teacher, and she guided 3 MSc students of PG School, IR in New Delhi, and her publications also, research articles are 24. And beyond that, book chapters, technical articles, manuals, proceedings, a lot of proceedings and manuals also there. And Madam, welcome you. And next speaker Next, Dr. G.D. Harish, scientist in the Division of Germplasm Conservation, MBPGR, and he is a curator of rice germplasm at Indian National Gene Bank, and he is rationalization and documentation of trait-specific information of rice germplasm. And he will monitor all the rice germplasm, and he will supply the material rice germplasm wherever the indenters required for research purpose or commercial purpose. He will assign the duties, total the management aspects of curator. And his publications are research articles, 16 articles are there, and several other book chapters and other things are there. And ma'am, these are the participants from 18 organizations, and from including SAUs and other non-ICR institutes also there. And please, ma'am, kindly share your experience and methods or methodology of seed viability and how to conserve and exclusively on cereal crops. And I hope you are the presentation will be very good for viewers. Please, ma'am, over to ma'am.
Yeah, thank you, Ravi.
Thank you, Ravi.
So before starting, actually from the participants, we would like to know how many of you are directly dealing with seed viability assessment? Are you, most of you, as has been informed by Ravi, are one way or the other dealing with PGR management. So in PGR management, are there people who have hands-on tried or have to try seed viability assessment? Any of you in this group in different crops, in any crop, are there people who are dealing with seeds in the context of any seed quality assessment in laboratories? Do we have anyone with working experience or require this information for their own working in their respective institutions?
Participants, please.
Since it is an afternoon session, we thought we will keep it interactive.
Yes.
If we keep on talking and nobody is there to listen, then there is no point in that. That is why we thought we will keep it interactive.
No, madam.
Okay, like nobody is there who is dealing with seed per se.
Madam.
Yes, please, Dr. Anuj. Yes.
Because we cannot see the audience video in full mode, only your voice can be heard.
Dr. Malar Vijay, Dr. Tangavendan, you asked already a lot of questions on seed conservation.
Sir, actually, sir, good afternoon to all, but my area is mulberries. We are using maximum that vegetative propagules only. That is cuttings we are.
Yes.
But very rarely we are hybridization. If done, then we are using for that mulberry seed. Whenever that closed hybridization work is going on, that time we collect the fruit and we raise the saplings from that seed. But it is less than 40 to 50% only seed will come.
Okay.
But if any technique like minor seed viability test is there, it may be useful for hybridization program.
Okay. So irrespective of your requirement or your level of requirement for your respective crops. So today when we are speaking about this, I think it will be, the information will be relevant for anyone who is handling seed. We will keep the discussion in such a way that the information conveyed will be aiding in a better understanding. It is, I do not want to keep it as a routine of how to spread the seed on the germination sheet and how you take the reading. That all of us know. That is not something for which any of you require training or any further understanding. That has been done. We have done it right from our graduation to your any post-graduation specialization. Every time one or the other, you would have done that. So we would stick to some very fundamental things which everybody as a plant science personnel in a research organization who's handling seed would need to know for their better understanding of handling of the seed. Okay, so the title is seed viability testing. So we will be speaking from the context of seed viability, because we all know the theme which is going on since the last 2 days for this training, we are dealing with conservation. So in conservation, the fundamental thing that we need to protect in the seed is the viability. Viability is our prime concern. And and I would say, like when we talk about conservation per se in the previous lectures, it must be already covered that why a conservation terminology is different from storage. How conservation is different from seed storage. Like that must have been already discussed. If any of you can enumerate on what is your understanding, how conservation and storage are different. There are experts sitting here, I very well know, who is knowing it better than me. But this is just to keep it interactive. Don't take it offensively that you're asking silly questions. This is to make it interactive so that nobody sleeps off or runs off in between the practical. So that I know that people are there. So how a conservation and storage is different from each other? If anybody can put in, then we can discuss.
As per my thing is a conservation, maybe it is a longer time, and that the storage, maybe it is a particular period, and for as and when required, we can. The conservation, it should be useful for the long period.
Okay. In a particular context, you are right. Longer duration, we always maintain conservation. We don't conserve for 1 or 2 years, definitely. But then this is not about duration alone. When we talk about conservation and storage, the primary difference, I mean, both we are concerned with seed quality. In both a good seed storage as well as in conservation, we should ensure the seed quality, maintenance of seed quality. But in conservation, we also, because in conservation, we are talking about the seeds as populations. Okay, we are conserving populations in gene banks, where we are conserving, we're talking about populations, and in that, the allelic representation of the population becomes very important. So genetic integrity and allelic composition of the sample we have to take care of is the additional thing we do in conservation as compared to storage. When you particularly store a seed sample, we can store a seed sample. It can be 100 seeds, it can be 200 seeds, it can be any, the seed lot size can be as per the requirement for which it is being stored. But when you conserve, you have to take care of the allelic composition, all alleles to be represented in such a way that the population is sound to be called as that particular variety or a genetic stock or whatever you are conserving. So you have seed numbers fixed because of that reason only. We say 2,000 seeds in self-pollinated crops and 3,000 to 4,000 cross-pollinated in order to take care of that particular requirement that we need to represent the alleles of that particular population in our conserved sample. In storage, that is not the primary requirement. That is whatever we have received, you maintain the quality. But here quality plus genetic integrity becomes a matter of priority. Now, when we are coming to conservation, today our topic is focusing on that seed quality parameter. We're not going to discuss about the allelic composition or how, but definitely interrelated, that I will explain in the course of the presentation. But the primary concern, our topic, is the seed viability, which for the seed technologist as well as a PGR person is important. So in order to maintain seed viability, the first step that has to be done is you start with a sample with the highest possible viability. So we do not compromise on the initial viability of any sample that comes into the gene bank, so that the long duration of conservation, we can ensure that minimum loss only happens. So initial viability is our prime concern. So we all know we do not keep seeds which is less than 85%, having less than 85% viability. Now, another thing which I need to discuss is viability and germination the same? We always loosely use the term viability, germination. Are they the same? Or can we use it interchangeably? Or is there any difference between these two? Can we say in any context, wherever we use viability term, can we use germination? Anyone who can throw some light on that? I think video is on only for 2 people. I can see more. Yeah, Anuj Kumar is there with the video on. So Dr. Anuj, can you throw some light on?
Yes, ma'am. Actually, we can say that it is finally interpretation that seed is viable through the germination. I think.
Yeah, you're right. So what happens is viability becomes a trait of the seed, and germination is the parameter which we use to understand viability. Yes.
Okay.
So in that, what happens is many times when you say this seed viability percentage, doesn't mean germination is the only criteria for viability. That is where I want to distinguish. Okay, we have other parameters also, but germination becomes a physiological parameter which we use the viability status of the seed. So a person who is totally out of context from the seed technological parameters, many times we see that the germination in all contexts, germination viability is interchangeably used, and many times it doesn't fit to be interchanged. So this we should understand that every time viability and germination is not to be equated. Viability is a seed trait for which we use the parameter germination for assessment, and because seed germination is the most easily and standard acceptable recommended test for viability, ascertaining the viability of the seed plot, we have laboratory germination test as the most easy and executable protocol for assessing seed viability, the most repeatable, reproducible protocol across labs with minimum facilities. So that is why that has become the standard viability testing procedure. Okay, so I will start sharing my slide, and we will go ahead with a discussion using the slides. Some share mode.
Full screen.
My slides are visible.
No.
Not, ma'am, not visible.
Minimize.
I think you've not shared.
Share screen. Yes, ma'am.
Okay.
No.
Now we'll go over here. Yeah.
Ma'am, it yes, ma'am, it is visible.
Right.
Yes, ma'am.
So I'm not going into some of these slides, is just for your reference, and anybody who is interested in specific points, we will be always available for further discussion. But keeping in mind the diverse group and a very like diverse subjects that we are there, for many people a very in-depth information will not be very relevant for certain aspects. Whoever is like wanting an additional point of discussion on that can always interfere and stop, or further later also, and another day also we can discuss. But otherwise, for everybody's, relating to everyone's specialization and concern, we will keep the discussion very general. So now this is something which we have all, a very preliminary information, what physiologically germination is. Okay, a diagrammatic representation, we all know once a free water is imbibed, now the enzymatic activation which happens, mobilization of storage reserves, how gibberellin is involved, how aileron layer in monocots is involved, and how the starch is broken up, sugar is transported, and embryo growth, the cell differentiation and tissue reorganization happens, and the essential sequence structures emerge. So that is what physiological germination is. Okay, this is just a recollection, a revisit to our student days. Now, as I told before, germination test is the one which is the most routine and recommended and reproducible test for seed viability assessment. And that is not the only one, but this is the most routine, and as recommended by International Seed Testing Association, we have standard protocols for laboratory germination test in all crops. And because the only reason being that this has the most highest reproducible character across laboratories. So we go for this test, which is easily done for all crops, and we have standard procedures for that. Now, in any germination test, we all know the recommended conditions. We standardize few conditions for each specific crop. One is the media, the other one is the temperature, light requirement, and this duration for which the germination has to be observed. These are the standard things which requires specification for each crop or each species. And we have the International Seed Testing Association that has standardized this, and in the gene bank, we follow those protocols for conducting this particular test. Now here, all of us are aware of such protocols. In either plain water we can use, or even agar medium we can use for conducting the test. When very small species like orchids, many times agar becomes more effective. And another thing is incorporation of nutrients and dormancy breaking chemicals becomes easier in case of agar medium, and even in the case of these where free water is used. Now, as per the International Seed Testing Association, the general recommended medium are paper, filter paper, and sand. And the different methods also we all are aware: top of paper, between paper, pleated paper, the sand, medium sterilized sand, how it is used. And these are all, I think one or the other way you would have done it or seen it also. Okay, so for small seeded species, we use Petri dish with the top of paper protocol. And if the larger seeded ones like wheat, maize and all, we go for between paper. And in those cases, sand is also recommended. And another important condition, in addition to media, what I told is the temperature and light requirement. So let us see in this context, like seed germination. So why we need to standardize all this is because seed germination is in general regulated by water, temperature, and light. Now, I would like to take you to a slight understanding about these three: water, temperature, and light. Because we all know, yes, we need not, while conducting the experiment, we have a ready reckoner with us. ISTA book is there. Just look into it, see what is the temperature, see what is the light requirement, see what is the duration, and just follow that. That is where. But in our, just to have a slight understanding about why this temperature, light, and all these requirements are there, we will have a slight discussion on that predominantly, because just to show you the medium and just to show you the basic testing protocol is, I think, irrelevant, since we will be already knowing that. Now, water in seeds, when water starts sorption, the water, the level, the water content, if you plot it against the relative humidity, as and when the seed increases its moisture content, we can divide it into three components of water within the seed: the lowest water percentage that is absorbed into the seed and the highest level of water sorption. Between that, the total water percentage in the seed, we can divide it into three phases. So the highest percentage, like when the water is at its maximum sorption capacity, there will be a lot of free water in the seed. So this particular free water level, if you can say, see 85, above 85% RH, you will have a level of free water. So for the seed to germinate, this free water is essential in the seed. The seed should be in contact with free water, so that free water is available. The RH of the atmosphere should have a lot of free water, so that sorption happens and the seed moisture content goes above, as you can see here, 25%. And only the presence of free water at this particular level, to this extent, will cause seed to germinate.
Heterogeneous fragrance.
Pardon? Okay, so lesser than, let us look at this middle level, 15 to 85%. We have weak or loosely bound water. We have weak or loosely bound water. And below that, we have the tightly, predominantly strongly bound water. So you must have in the previous practical classes discussed about seed drying and all. Now, when you dry for conserving the seed, what you do is we remove the phase 3 and phase 2 water, and you keep the seed in the phase 1 water. Okay, that only the strongly bound water should be there for ensuring long-term conservation. So because what we do is that even the presence of the phase 2 water will ensure a certain level of metabolic activity. So when you have to minimize the metabolic activity, you take out phase 3 and phase 2 water. And only when you re-imbibe and bring the water to the third phase, then only the seed proceeds ahead for germination. Now, the second requirement was that of germination temperature. Now, if you look into the ISTA germination chart, you can see that the requirement varies. And we all have a very fair idea that these winter crops or the rabi crops or temperate species, they all have a lower temperature requirement of, for example, wheat has a 20 degrees centigrade. Summer crops, tropical species, about 25 degrees centigrade, sorry, 25 to 30 degrees centigrade, like that. So that is based on the performance. If you can see the chart, all these winter plants, the maximum germination, the optimal germination percentage values is around towards the lower side in the case of these plants, whereas it is, that is how the seed features have evolved. It all during the natural evolution based on the seeds temperature regulation or the metabolic activity regulated by what optimal temperature. That is what defines the appropriate temperature of seed germination. So we have the ISTA has fixed the temperature standard germination of crops based on their natural acclimatization over the period of evolution to the best optimal temperature. That is what decides whether wheat should be kept at 20, that gives the best assessment of viability, and maybe paddy at a higher temperature gives a better assessment of viability. All that is fixed based upon this data. Now, we all know that in the case of temperature in the natural environment, when seed is sown in the soil, it is not a constant temperature which the seed comes upon. In the lab, we are giving 20 degrees centigrade for wheat. We are keeping it in the fixed germinator with 20 degrees centigrade, but that is not what happens in the soil, right? In the soil, we all know near the surface, there will be a higher fluctuation, diurnal fluctuation, which we call it, of alternating temperature will be happening between day and night, whereas it will not be much in the case of deeper sown seeds. So this is also taken into account when we fix the germination parameters. And we can also see whoever has gone through the ISTA protocol in the standard germination table for some crops, especially for small seeded species, you have diurnal temperatures fixed, because as per their natural acclimatization, they require this exposure to diurnal temperature. The stimuli of diurnal temperature is required for natural germination to happen, and that is what is replicated or simulated in our laboratory germination test also. And where in such crops they are sensitive to light and small seeded, you can see that in the chart they have written diurnal temperature. So the best laboratory results which we can relate with the planting value of the seed lot is obtained only under those conditions. So if you have to test a seed quantity and ascertain the nearest possible or the best possible value, planting value of that particular seed lot. The basic purpose of conducting the viability test through germination is to assess the planting value of the lot. So in that, all these conditions, simulated conditions we should follow, then only the best reproducible results can be obtained. Another one is the effect of light. Now, what is our concern with light? How light influences germination? Now, we all know seed has the chromoprotein called phytochrome. And this phytochrome, as the name itself suggests, it is light sensitive. Now, red light, when red light is perceived by the seed, it causes the phytochrome to change from its inactive, biologically inactive form, which is the PR form, to the far red active form, PFR form. So PR is biologically inactive and PFR is biologically active. So once red light, PR is the phytochrome condition form where it is sensitive, where it can perceive the red light. So once after perceiving the red light, PR shifts to PFR. And PFR is the active form. So only once the PR, if the PR is predominant in the seed, it will abstain from germination. When red light is perceived and it proceeds shift to PFR, then only the seed will germinate. Now, what is the relevance of this PR and PFR and the natural germination conditions for the seed? Why this particular light? And it is, and we put these standard light conditions for germination, which is recommended because based upon the light sensitivity and this PR, PFR sensitivity, even in our laboratory test also, the light requirement is based upon the sensitivity of the seed to this red light and far red light. Now, what happens is why this sensitivity and how it helps in germination, helps in the deciding the seed to decide whether to germinate or not under natural conditions. That we will see. Now, under normal sunlight, the ratio of red light to far red light is in the ratio of 2:1. So what happens is whenever the natural sunlight is perceived by the seed, there is a higher amount of red light in that. So the seed tends to shift to from PR to PFR. So under natural sunlight, under normal conditions, if the seed is sufficiently exposed to sunlight, the tendency is always to germinate from PR to PFR. And we all know it requires a water and temperature conditions additionally. But from light, natural sunlight provides the sufficient red light to shift the PR to PFR. Now, if it is under a foliage canopy and a forest cover, where there is thick foliage above, what happens is far red light, due to its higher wavelength, it will have a higher penetration effect through the foliage. So that far red light will be dominant. Far red light which reaches the forest soil through the canopy, penetrating through the canopy, that will be more dominant as compared to the red light. So whenever the canopy is thick and only far red light is reaching the soil, so that is an indication or a message or a trigger for the seed to remain ungerminated. only when there is space. So that it's what it indicates is that it is a very crucial survival mechanism for the seed, light sensitivity. Once it emerges and then there is no sufficient sunlight for photosynthesis, the seedling will perish. So this particular one determines the survival mechanism of the seed, in fact. Same as when it comes to the depth of sowing. Under very deep sowing, far red light only penetrates. And here again, the signal is not to germinate. And whenever there is tilling of the soil or whenever the seed is exposed to light, there it immediately sprouts. So that is how, in fact, the soil seed bank survives also without germination. And immediately, if you can see in the case of weed seeds, weeds with smaller seeds, in general, all small seeds have this extreme light sensitivity, and they retain their dormant status until their phytochromes get converted to PFR from PR through the perception of sunlight, and that determines their survival. Why? Because once germinated, they do not have nutrition enough to ensure their survival. So what happens is they are clearly dependent upon photosynthesis right from immediately after germination, because their nutrient capacity is not sufficient to prolong survival of a seedling for any number of days. So now, unless sunlight is there, they will perish immediately. So that is why small seeds are extremely sensitive to sunlight, and even the seed bank, the soil seed bank, the seeds that are, natural soil seed bank species, they all survive through this perception. Weed is the best example, weed species. So we were discussing till now about these parameters, these standards which we do, or what is the principle behind fixing these standards for conducting the germination test. Now, once we conduct the test and we got it on our germination medium, be it sand, be it the filter paper, be it the Petri dish. So in that, we will look into the sample. Suppose we had put 25 seeds, and then we are taking out the Petri dish, and we know the day at which the Petri dish has to be opened for counting. In the case of wheat, 7 days. In the case of paddy, 14 days. In the case of pulses, many pulses, 7 days. So like that, on that particular day, we opened the Petri dish. Now, we all know that the percentage has to be seen. So when we see the percentage, we look into the categories of seedlings. We have the normal seedlings, abnormal seedlings, dead, and fresh ungerminated. Okay, these are the categories which we need to look into. And always the percentage is counted based upon the I'm waiting for answer from any one of you.
From normal and fresh ungerminated or partial.
Thank you. Thank you. Your doctor. I just couldn't get your name. Your name, please.
My name is Dr. Hridesh Anuragi.
Okay. So you are from where?
ICR Kafri, Central Agroforestry Research Institute.
Okay, yeah. So thank you so much for speaking up, because I can see most of the participants, I think, have left. So in this germination test results, as Dr. Hridesh correctly told, normal seedlings we will be accounting and fresh ungerminated. Now, in this, but it is very important for us from the gene bank perspective to note the abnormal seedlings also. Why? Because for a seed technologist or a person who's immediately going to the seed to sow the seed, or for a breeder, maybe abnormal seedlings may not be of that relevance. But for a gene bank curator to note down the abnormal seedlings is very important, because it is the first indication of seed deterioration in the seed, in the lot, in the sample which we are going to conserve. And any occurrence of a high percentage of abnormal seedlings is indicative of an already deteriorating seed lot, which may not be accounted or estimated when we just count the total seedling percentage. Because normal, if the essential seedling structures are all normal and visible, we count it as a normal seedling. And in that, what happens is, if the abnormal seedlings are there to a certain extent, that means the seed lot has a certain level of deterioration happening, and the desirable conservation period may not be obtained in that particular case. So if there is a high number of abnormal seedlings, it is always advisable to not conserve it and get it regenerated or replace it with a fresh sample. Now, because for dead, we will be anyway be cautious, but for abnormal also, for PGR, for a person, gene bank curator, we should be very alert about abnormal seedlings also. And we all know ISTA as its definition of abnormal seedlings. Some slides I will show as some examples. We should be very clear about how to differentiate a normal seedling and an abnormal seedling in our respective crops. Then only our eyes should be trained for that. It is not a very easy thing to find out from the petri dish when we are evaluating thousands of accessions per day. But then our eyes should be trained for that, so that when you conserve a material for 50 years, you know that the material is properly conserved. Now, fresh, ungerminated, and dead. We need to distinguish that, because in both cases, the seed has not germinated. Okay, so in how to distinguish that, a dead seed and a fresh, ungerminated seed. If any other person can contribute to this. How do you think in the petri dish you can differentiate a fresh, ungerminated, and a dead seed? Anyone who has an experience of handling a germination sheet, or just out of
an
common
abnormal
logic?
seedling?
Yeah.
Excuse me. Yeah, an abnormal seedling will have a little bit of radicle or tubule coming out.
No, we are
but it
talking
won't
about
grow
dead and
properly.
fresh ungerminated. How to distinguish dead seed and fresh ungerminated seed?
Dead seed, you won't see any swollen part. That means it won't imbibe any water also. But in a fresh and ungerminated seed, you can see some imbibition of water, but it's not germinated yet.
Yeah, so this is exactly what I wanted the particular participants. I was expecting that this reply, but what I would say is that always a dead seed will imbibe more water than a fresh ungerminated seed. A dead seed will be more swollen than a fresh ungerminated seed, but that is not going to be our criteria. It is not a distinguishing criteria. Why I told dead seed will imbibe more is many times it won't be visible outside that it has imbibed more. For distinguishing purpose, we cannot go to use the level of imbibition as a criteria for distinguishing both. The only way in which we can distinguish the dead from fresh ungerminated is slightly press the seed. When you press the seed, if the seed is soggy and not firm, it is dead. A fresh ungerminated seed will be always firm when you press. So that is the first test you need to do in order to distinguish both. And then afterwards, when you have a large number of fresh ungerminated seeds, you go for the cut test to see the firmness and distinguish the dead. Now, why I told the dead seed imbibes more is because once the seed is dead, the seed does not have the counter turgor pressure. Imbibition, we all know, is a physical process, and it is determined by the metric potential of the seed solute inside. And always when the seed imbibes, there is a counter pressure from the cell wall to it is an opposite reaction from the cell wall. In a dead seed, that is not there. The counter pressure is not there. So imbibition is always higher. The dead seed continues to imbibe. Is higher, but it need not always be visible as a more swollen one outside, because there will be more leaching, or there may be different kind of this thing. So we cannot assume that a dead seed will be more swollen or a fresh ungerminated will not be swollen at all. No, you cannot go for the swollen criteria. Just press it. If it is soggy, it is dead. If it is firm, it is fresh ungerminated. Now, a fresh ungerminated seed of large percentage, you cannot just leave it on the petri dish and then declare. We need to confirm its viability. Now, for confirming its viability, before that, we were talking about abnormal seedlings. So in abnormal seedlings, I just have some few examples in order to show you the kinds of abnormal seedlings in different crops. So wherever you have spinkly growth of the shoot or root, geotropism is not properly negative or depending upon the requirement. And if it is a glassy structure, if the seminal roots, if it is the species has seminal roots, and if it is less than 2, especially in monocots, then I'll show you the example of wheat also. And then if the coleoptile in the case of monocots is cracked more than 1/3, if the leaf structure is not emerged, any kind of these things, there are clear-cut definitions in ISTA. It is not that you see and then you assume that based upon the structure. No, we have clear-cut definitions. In the case of cotyledon species, cotyledon should not be glassy or decayed with primary infection. You should distinguish between secondary infection and primary infection. So only if it is a primary infection, we categorize in the abnormal category. So all these things for your crop, we should be very clear. And the standard definition of abnormal seedlings we have to follow. We have to identify and label it specifically as abnormal seedlings. So once we had after abnormal, we had come to dead and fresh ungerminated seed. So when the fresh ungerminated is very large, we do the cut test. Now we find that, yes, they are mostly in this particular seed lot, fresh ungerminated. So in that, what happens is we need to establish its viability and then include that in the viability percentage. All fresh ungerminated, okay, lot of that is there, just count it as normal and declare it. No, it is not like that. We have to ascertain its viability. So wherever one method, the shortcut protocol which we use is the tetrasolate test. So I'm not going to elaborate it because you all are aware of the protocol, a very simple test where a viable seed tissue will have the dehydrogenase enzyme activity, and we are giving it a suitable substrate so that wherever the dehydrogenase enzyme activity is there, it will cause a particular enzyme substrate reaction, and the product formazan, which is this particular colored product, will be present only on the live part of the seed. Why? Because the dehydrogenase enzyme activity is only at the live part. So the live part is indicative of an alive tissue, and the discolored part or the uncolored part where pinkness is not there or the redness is not there is the dead part. So this particular pink color is indicative of the live tissue. Using that, we call it topographical tetrazolium test, because the topography of the staining pattern is what we are going to use for deciding the viability of the seed. So all the essential structures, the embryo, if it is sufficiently pink, there we go for. And in this particular one, we can see the aileron layer. So for every seed, it is defined. We need to define what is we need to know the seed morphology in such a way that we know what is the seed tissue that is expected to be colored. And to what proportion, ISTA has again defined the proportion to which that particular coloring should be covering the topography so that we can label it as a viable or a non-viable one. Okay, so for viability assessment as a quick viability testing protocol, we have this. So whenever there is a high degree of dormancy or a fresh ungerminated seed which is present, mostly because of physiological dormancy, then we do this test and ensure its viability, and then we include it in our viability percentage. So here I have brought some slides to show the difference between the viable seeds as per TTZ test and the non-viable seed. So in the below one, you can see that the embryo portion, many of the essential structures which will be required for developing into the radicle, in the root and the shoot, they are not colored, they are dead. So they will be easily going into the viable, non-viable category, whereas in these cases, you can go into, we have the real viable tissues. Again, for cotyledonary seed dicots, viable, and here you have the non-viable. See, the entire radicle is, though the other portions have taken color, a crucial structure of the seed is not colored, so we have to categorize it into the non-viable category. So in this, knowing the structure of the seed clearly is very important. You should know where the essential structures of the seed are, then only you will be able to assess the viability topography. Again, this will be how, if it is totally dead, no color will be taken up even after conducting the exposing to the tetrazoa. Okay, so now I would be little into these water uptake, which we were talking about. We were talking about imbibition and about the take up by dead and the germinating seeds. So whenever for the germination, the water is taken up, Ravi, we have till what time we have to complete?
Ma'am, 4:30, ma'am. No, ma'am, you can.
Okay.
You can extend time also, ma'am.
No. It is not required. I was thinking like it is 3:30 or 4:30. I just wanted to confirm.
Yes, sir.
Okay.
4:30.
So, yeah. So when we are discussing about germination of the seed, the basic, we discussed about the moisture uptake. We discussed about the temperature as well as a light requirement. So let us look a little more into these conditions from the principal perspective. Okay. And here we know, everybody knows the germination, water uptake, it occurs in a triphasic pattern. And this triphasic pattern, this is what I was discussing about. Even in the case of a dead seed, it proceeds in such a way that the below blue line is the dead seed, because there is no counter turgor pressure which can stop the uptake of the seed. So that is why this continues in this manner. So in order to distinguish the dead seed from the germinating seeds at this particular stage, at phase 2 stage, it is not possible. But once it reaches the phase 3 and reaches the soggy stage, then you can see it visible in your germination test. And again, another important thing which anyone handling the seed in majority of species, for example, like soybean, anybody who's handling soybean seed, they will all know when a very dry seed which has been dried to less than 7% during conservation, we all dry the seed to less than 7%, at times to 5%. Okay, so those seeds which are at 5 or maybe 4%, if you directly put it to free water, because for germination we need to expose to free water. At times, what happens is in many of the genotypes which are having very thin seed coat, imbibitional injury happens. Rupture of the tissues happen, and because of which what happens, the viability is lost at that particular time. Maybe radical emergence will happen, but it would not be established itself into a full seedling. And it is more severe at lower temperatures. So in order to prevent imbibition damage, this protocol can be followed in whichever crops you people are dealing, and you face imbibitional damage while trying to germinate the seeds. So you use this protocol of holding the seed above water in a sealed box at 20 degrees centigrade. What is happening is you are giving a 100% RH to the seed. Okay, so the seed is coming to a very high, it is not in contact with free water, but it is coming in contact with 100% RH. So it will equilibrate to around 20 to 25% seed moisture content. So once the seed cells are equilibrated to 20 to 25%, then you expose to seed free water, the imbibitional injury will be minimized or avoided. Okay, so this is very essential in many of the crops, even while in laboratory we are assessing. In crops which are prone to imbibitional injury, if you try to do directly, you will get wrong values. So you have to use this protocol, and then only you should go for assessment. That will give the real viability assessment. Now, a little I would like to discuss on, we are all talking about seed viability assessment, and in gene banks, we do not use viability assessment just to find out how is the seed which is coming into the gene bank, whether it should keep it or not keep it. No, the viability is not just for seeing whether it is above 85% initial viability. We also use these viability value for monitoring the performance of that particular accession in the gene bank. And we also have protocols for using the initial viability value for predicting its storability. So by estimating the initial viability status, we are estimating, or we can even try to predict the storability of the seed lot using specific statistical tools. And a very basic thing which seed technologists all use is the seed viability equation. And within the seed viability equation, we all know that in the seed viability, it is not that initial viability alone plays a role. It is always viability. It cannot stand as an independent entity. It has to be related with moisture and temperature of the storage environment or the condition in which the seed is exposed to, the relative humidity and the temperature parameters. So its relative humidity automatically translates to seed moisture content. So that is how, while predicting, so we need to take into account all these three parameters: initial viability, moisture, and temperature of the seed, or the conditions in which the seed is exposed. So I would like to give a brief, outlook into exposure to how that is done, a viability assessment, and how the prediction of seed viability is done. So what I have depicted here is the basic seed survival curve. Seed survival curve is nothing but the performance of germination of the seed, or the seed lot, I would say, over the course of storage. So it follows this inverse sigmoid. And because we all know the germination drops during the course of storage. And as soon as the germination drops to 85% of its initial viability, we go for regeneration in the gene bank. Okay, so it is a very important activity for which we keep on monitoring the samples. We monitor the seed viability after conservation, in a regular interval of 5 years in the case of species that are poor storers and 10 years in all other species. So after 10 years, when you take out the accession and put out germination test, all that is done through simple germination test, viability assessment. So when you take out and do it and find that initially it was 100% and now it is 85%, then what happens is it has slipped to less than 85% of the initial. You mark that accession for regeneration and take it out and multiply and keep it back. Now, why we are doing it is because the initial seed quantity, because we are conserving, it is not just storage. So we are keeping 2,000 individuals with the assumption that all the individuals are contributors to their allelic diversity, the representatives. And the number is fixed in such a way that it is capturing the rarest of alleles in the population. The number is fixed in such a way that we are ensuring that the rarest of alleles in the population is also represented in our accession. During sampling, we have covered that also. Now, when individuals start losing, when viability is lost from 100, it has come down to 85. That means you're losing 15 individuals from the population of 100. So that means the first alleles to go out will be the rare alleles. So you're losing the allelic composition. Genetic integrity of the accession is getting lost. So that we cannot afford to when we are conserving a sample. So that is how wherever we are keeping accessions, it is not that initially every accession will have 100% initial viability, right? There may be accessions because anything above 85 we are going to keep. Though 100 is our target, if you have an accession coming in with 90%, that is also conserved because that is above the threshold. So in 90, what we look for is 85% of 90. 85% of 90. There, when it comes down to that particular level, then only you will take out. Everything is not taken out at 85. It is 85% of initial viability. Okay. So there you take it out. Now that is about regeneration. Now, when it comes to predicting final viability, we have this seed viability equation, which we use to predict. Now, in order to understand the seed viability equation, a little principle behind the seed viability equation, we need to understand a little more about this application of the seed survival curve. Now, we all know in order to statistically predict using mathematical equation, linear mathematical equations, this sigmoid curve is not going to function. So that is why we convert our germination initial percentage value to this probit values, because a probit curve will be always a straight line curve. And this probit viability values, now when you plot it in the same manner, okay, the predicted values in the same manner, then what happens is you will get these slope as 1 by sigma. Sigma is a horizontal line which is shown here. Now, what is the sigma? Sigma is simply the time taken for viability to drop by 1 probit. And inverse, it will be the inverse of slope, because 1 by sigma will be the slope. So that is going to give us the sigma. So sigma becomes the prediction value in the context of the seed viability. Now, what happens is, what happens to the sigma value? We all know, if you consider, let us consider the moisture and the temperature components separately. First, how moisture is going to influence the sigma, and then how the temperature is going to influence the sigma. And finally, how we will incorporate both and develop how the original seed viability equation was derived. That is what we are coming to. So in the case of moisture, we all know that higher moisture content viability is lost faster. So we will have the sigmoid curve landing up near to the y-axis. Similarly, it will be keeping on increasing towards the lower moisture content. If you get 10% moisture content, you can see there is a more indefinite period of storage. So when you convert it into its respective probit values, the below graph will show the respective probit graph. So in the case of 10%, you know, we have the highest sigma. So what is sigma? Time taken for viability to lose by 1 probit. So whenever substorability is more, sigma value will be more. Time taken will be more. Loss of viability is happening after a longer duration. So that is because of which sigma is a higher value here. So likewise, we can, based on this, a relationship can be established between viability and initial viability percentage and sigma. So we have established, initial viability percentage and sigma in relation to seed moisture. Okay. So once that relationship was established through multiple standardizations, next one was, this again is how they have derived the linear relationship in terms of seed moisture content and storage period. And here, in order to derive a statistical equation, again, the profit values were converted to longevity values, and we have derived this particular log sigma equation, connecting the moisture content with the sigma value. Okay, and this particular relationship functions only in between the relative humidity of 10 to 15% and 85 to 90%. Beyond that, it doesn't follow a linear relationship. So a viability equation will function only between these range of relative humidity values. Okay, when seeds are equilibrated or the EMC of the seed is in relation to these RH outside, then only the seed follows this particular pattern of viability prediction. Similar temperature and storage life. Okay, so again, using these statistical parameters, we have established a relationship between sigma and temperature. So the sigma, which is the time required for probit viability to drop by 1 unit, in that, in terms of temperature, we could establish a relationship using this longevity scale. And here, if you can observe that here from 0 and here between -40, we have -20. So if you observe closely, we can see that till -20, there is here, if you can observe here, as and when your temperature is near 0, and at lower temperatures, the viability is at its peak. And at -20, it is having the highest value. But if you reduce it further, it only dips. So what I mean to say is, when a gene bank fixes the temperature of long-term conservation as -18 to -20, this is the logic behind it. An orthodox seed that is having a moisture content in a particular range of equilibrated with 10 to 15% or 20% ERH, which obeys the rule of sigma moisture correlation. Here, if you keep it at -18 degrees centigrade, that will have its highest viability. It is under that logic that we have fixed the temperature of our long-term storage modules at -18 degree to -20 degree centigrade. Now, once the sigma's relationship with moisture and temperature have been fixed independently, they were all put together into this seed viability equation, the proponent of which is Roberts. So we call it Roberts viability equation, where initial viability, moisture, and temperature information is inculcated to predict the final viability. So this equation, when it is solved, it will give the final viability after time t. T is the days of storage. So that is how viability assessment is used in gene banks for predicting the viability of accessions. Now, this is actually, we were discussing about physiological parameters, right? But we all know viability in terms, there are several components of biochemical metabolic components which are regulating seed viability. So definitely there must be parameters which are indicative of their viability, of seed viability statements, like status, in addition to the germination. Germination is a physiological expression. It is a physiological parameter we are assessing. So, but what about the biochemical parameters? Are there any parameter which can reliably and reproducibly give the viability status? The principle which we are using for this particular understanding is, we all know in any living cell, be it human or any animal or any, we have a gene as a core mechanism happening due to free radicals. It is the free radicals continuous reaction, the chain of reactions triggered by free radicals on our biomolecules in the cell that is causing the cell to age or deteriorate. And this particular is a process of free radical attack is countered by our antioxidant enzymes. So a cell which is at a younger stage will have a very established antioxidant mechanism. And whereas in an aged cell, this antioxidant mechanism will be weak. So the status of the antioxidant system is hence an indicator of the age of the seed or the level of deterioration of the seed. Now we can use this effectively in establishing the seed viability status or the extent of seed deterioration. Now in this, I've just given the categories here. The primary enzyme which we are using for that is the non-enzymatic antioxidant glutathione. Now, glutathione, when we are using it, one thing we should understand is all enzymatic assessments are basically qualitative assessments. Like we do not have a value to state that a GSH of this particular units, say 2 units, 3 units in a seed is indicative of the fact that it is 100% viable. And if it is at this 20 units, it is only 50% viable. No, it is not done like that. In the case of enzymatic assessments, what we do is we do qualitative assessments to compare between seed lots, which is better than this or not. That is the basic idea we take when we do these enzymatic studies. But in our case, in gene banks, if you need to assess a viability status, that would not suffice. That is not our like sole requirement. We need to have a quantitative assessment. So in that, what we do is, we all know about the basic function of glutathiones, what it does. So in glutathiones, what happens is glutathione basically, it happens in seed, in a regular seed, which is vigorous. It stays in its reduced form. Now, this is the basic free radical reaction. What happens is when the OH free radical, hydroxyl free radical, which is the biologically most detrimental free radical in our system. So this hydroxyl free radical is preferably, it's taken up by the glutathione anion, and that gets converted into the glutathione free radical. So this preferential, quenching of the free radical by these antioxidant enzymes is the core to the function of the antioxidant. Instead of the biological molecule, when glutathione is present in sufficient quantity, it occupies this free radical by converting, and in the reaction process, it gets converted into the glutathione free radical. Now, 2 glutathione free radicals combine together and form the glutathione disulfide. And this glutathione disulfide gets reverted back to glutathione. That enzyme is, that reaction is shown below by the glutathione reductase in the system. And again, it is replenished. The GSH is now again available for free radical quenching. So what happens is the ratio of this oxidized glutathione to the reduced glutathione, or vice versa, the presence of the reduced form of glutathione in the system is a very good indicator of the liveliness of the seed, degree of aliveness of the seed, viability of the seed, or low deterioration of the seed. So what we do is we go for this ratio, GSH to GSSG. That is one way of establishing. But that again is not fully quantitative. Again, the ratio we have to see for, we do not have fixed values for that. Which value is going to be giving a better estimation of viability. So what happens is, we use this particular value in the Nernst equation. Nernst equation, we all know in electrochemistry, we use it for calculating the half-cell redox potential. So in this half-cell redox potential equation, we substitute the GSH and GSSG values. And then what happens is, it has been reported, you please focus on the last paragraph. So generally, the viability percentage declines to 50% when the redox potential rises to between -180 to -160. Redox potential is in negative values. So we need to standardize for different species. If the electropotential value is between -160 to -180, what happens is there is a drop of 50%. And in between, so seeing that, we need to assess, we need to fix the range of viability for each drop in electropotential values, and then ascertain what was the initial viability, and now it has come to this particular one. So how much is a related viability drop? So that will consume lesser seeds, and also it becomes a more precise tool for predicting the seed viability. Okay, so that is one biochemical predictor for seed viability, which can be effectively used. This was basically, from the human system. In many cases, the pharmaceutical industry also uses it, and we have borrowed it from there, basically. Now, another test, which will be, the first basic test to see in the case of crops that are highly susceptible to borer attack and all. So before doing the germination test, we can do the X-ray test. And when infestation is predicted to a high amount, instead of wasting time and resources for this thing, if you have the X-ray facility already, scan it and see the level of this thing. If above permissible level, no need for going further. For further tests, just discard the sample through this only. Okay. And in tomato, it is shown here not for pest damage. Why it is shown here is, tomato seeds have always the problem of underdeveloped embryos. Okay. So if you do the germination test, you will find that many fresh ungerminated seeds per day, large number. That is because tomato seeds, the requirement of after ripening, or when you harvest as from the umbelliferous species, the secondary and tertiary, what do you call it, the branches of the umbel, there will be a lot of seeds coming in with underdeveloped embryos. So in that, what happens is they're not basically, they require the further after ripening period, and seed technologists go for priming treatment specifically in tomato or even in carrot. It is there. They are high value crops also. So in that, priming becomes very effective because of this. Priming causes the embryos to develop uniformly to the same extent, and then you sow it, it gives uniform germination. Without that, if you sow it, there will be non-uniform germination in the field, which is not feasible in the case of crops like carrot, where machine harvesting has to be done in a single go in the field. So in this case, what happens is, again, a scanning with the embryo will give us an idea about the amount of fresh ungerminated seeds, and accordingly, you can plan your experiment or sowing. Now, in addition to seed viability assessment, we all know vigor of the seed is also a component which is indicative of the viability or the level of deterioration. Okay, not the immediate planting value of the seed lot if you say the germination is sufficient, but if you have to conserve and if you have to know the progress of deterioration, vigor becomes a very good estimate. And what happens, the graph what is showing here is that the vigor of the seed collapses much earlier than the viability. The total plant, the normal seedling value may be quite same for two seed lots, but the vigor of the seedlings may be quite different in both seed lots. So after 10 years, if you take up, viability percentage would have dropped for the lot where the vigor was less, but it may be still maintained. Viability may be maintained even after 10 years in a seed lot where the vigor was good. So estimation of vigor is always important, but the thing is, it is not routinely done in any gene banks because we cannot expend seed material for that. But wherever we have a doubt that this particular material is not good enough, is not vigorous at all, and there is no point, after 10 years, it may have a severe loss of viability. So it is always advised you do the vigor test along with the viability test and ensure proper viability. If it is not at all vigorous, if the vigor estimates are very poor, again, discard the sample. Okay. So one test which we do for that, just one test we will discuss here is a conductivity test, electrical conductivity test. So we all know, as I told during the free radical reactions, when biomolecules get compromised due to free radical attack, one being our phospholipids in the cell membrane. So what happens is the structural integrity of the cell membrane gets affected, causing the osmoticum to leach out. And once you soak the seeds, the deteriorated seeds or the seeds which are having large number of free radical attack without much of quenching by antioxidants, they will have more leachates in the leach out. So estimate the leachates using electrical conductivity meter. Just see the conductivity of the leached out solution. A higher conductivity is indicative of more leachates, and they're comparing. Again, this is qualitative. You look into, compare the samples. So there, keep a standard fresh sample and then compare to see if it is a very high leachate which is coming out, that means its deterioration has set in. Maybe it will germinate. Your normal seedling count may be good, but a high leachate is indicative of poor seed quality. And in the case of Garden Pea, ISTA has standardized the values also. So you can use it as a quantitative protocol. In the case of Garden Pea, they have standardized the values. The protocol is fixed. How many seeds, how much soaking, temperature, everything is fixed. Once all that is done, you can use this chart to understand whether the seed quality is reliable or not. So that has been fixed. And that is very important for conservation also. Okay, so I think I have this much only to discuss. I'd be happy if now we can open up for discussion.
All participants are requested, please kindly interact if you have any doubts.
Any doubts or any point of, have I unshared my?
No, ma'am. Still is there.
Still it is sharing?
Sharing, yes.
Okay, yeah. So I've stopped sharing. And anyone who wants to know anything further about this, Anyone who had sat through the presentation?
Kindly switch on your videos, please.
So Dr. Deepti, you're working on which crop?
Yeah, I have just finished my project on dry flowers, and soon I'll be starting with marigold breeding.
So you'll be dealing with marigold seeds.
Yeah.
So you've not yet started, but.
No, actually I did it in PhD, then it discontinued for another 8 years, and there I work for dry flowers only.
Okay, okay.
Soon I'll be starting either on marigold or dog flower.
Okay. So when you're handling these seeds, you're going to get a lot of issues on germination. They're not very comfortable seeds to work on.
Right.
So, but you have ISTA has standardized for that. But it depends upon, then many times what happens is you need to have proper germination chambers with maintained temperatures, and you come across certain features of the seed which may not be replicated in the field. So that time you come across these issues. Kriti is working. Kriti, you're working on which you had experience in wheat, and now you're working on which crop? Kirti Rani. You're muted.
Doctor.
You worked on wheat, no, earlier?
Yeah, my PhD is on wheat, ma'am.
Yeah, now
Ma'am,
you
thank
are
you so much for
handling
such
multiple crops.
lecture. Ma'am, I had to ask, ma'am, if we have seeds of different crops in the storage, long-term storage, and if we simply what we do is many times we do the germination test for those seeds. If means in the field we do that germination test for those seeds stored for a long period of time and which have not been regenerated. So, ma'am, can we directly go for the viability test? Means how it is related? If it is viable, then means it will germinate.
You mean when you conserve in the MTS in your module?
Yeah.
And then if you have to take it out for regeneration, first thing you should do is take 25 seeds from the MTS sample, do the lab test, and ascertain its viability, how much viable it is. And then only you take out the sample. And if you're finding that viability is very low, you have to fix your seed rate depending on that, right? The number of rows that have been sown, so that again, in the case of your sample, you get sufficient seeds back. So it all depends. So if your viability has dropped below 50%, and then you are taking out only 25 seeds into the field, a single row to put for germination, you will not get sufficient seeds. So all these for determining lab test becomes very important to find out.
So before directly going to the field, we should go for the lab test, viability test, and then we can go to the field test. Means like
In case where sufficient seeds are there, you have large number of seeds in the MTS, yes, fine. You do it in the field with larger number of seeds if you can afford to to consume large number of seeds in the field itself. But where the number of seeds are less, most of the germplasm it will be, especially if you have land races and all. So the seeds won't be in abundance. So in such cases, you need to do a lab test first and estimate, and only when essential, you need to take it to the field for regeneration.
Ma'am, how much viability test and germinability test is related? Practically, I want to know. If they are viable, then what?
Yeah, Ravi?
Yes, ma'am.
Kirti has not heard the total presentation at all. उतना तो मुझे बहुत clear हो गया. वो पूरा presentation में से कुछ भी नहीं सुना है. अभी पूरा रामायण होने के बाद पूछ रहे हैं कि viability और germination में क्या फर्क है. आप ही बोलिए. आप IRI के student हैं और इधर ही बोले. आप ही बोलिए viability और germination में क्या फर्क है.
नहीं मैम, प्रैक्टिकली मैं जानना चाहती हूँ कि अगर हमने वायबिलिटी टेस्ट किया है और इट इज बियॉन्ड मतलब कुछ परसेंटेज है लिमिट, तो फिर वो जर्मिनेबिलिटी में कितना होगा?
थियोरेटिकली मुझे सुनाइए पहले, प्रैक्टिकली होने से पहले थियोरेटिकली बता दीजिए जर्मिनेशन और वायबिलिटी में क्या फर्क है. हम प्रेजेंटेशन ही उसी से शुरू किया.
वायबल दैट सीड्स आर अलाइव और डेड मींस
या तो जर्मिनेशन क्या हो गया?
Ma'am, germination, that's what I know. Apart from the if seeds are viable or not viable, other factors जो है जो means dormancy भी होता है, तो वो कितना मतलब related है?
मैं आंसर तो बता रही हूँ, but in the condition that रवि कीर्ति के absent लगाएंगे इस practical session की, ठीक है? उसी condition में मैं आपको आंसर बता रही हूँ. इसी में the presentation started with this particular topic. See, viability is a trait of the seed, है ना कृति? Viability is a trait of the seed. Yeah. And उसी को assess करने के लिए हम जो physiological parameter जो use कर रहे हैं वो germination. So germination is not the only means by which we can look into viability. But why then we are all running after germination is because that is the most easily executable and reproducible methodology we have, which can be done in any labs. आप एक remote area में बैठ के आपके पास एक बोरा का टुकड़ा भी है, उसी को लेके भी तो आप germination test कर सकते हैं, उसी से भी आप आपका planting value assess कर सकते हैं, है ना? तो that is why जो organizations जो standard recommendation करते हैं viability test के लिए, they go for those tests which can be reproducible and done with minimum resources. So germination test is the one which is going to give the, and why germination test? Because germination test is one where we can look into all the essential structures that are going to be giving a strong or healthy plant in the field. So that means we are assessing the planting value. So for any person dealing with seed, planting value becomes the most important thing that needs to be assessed. So planting value का direct estimate भी देता है germination test. So that is why for all these reasons हम viability को germination से equate करते हैं. Viability is the trait of the seed and germination is a physiological parameter which we are using to assess that viability. And we discussed other methods also which we can use, but germination becomes that one. So whenever आपको, because you are handling germplasm, when you have to take out the material germplasm seeds को आप जब मर्जी इस साल में तो कुछ काम नहीं है, थोड़ा सा regenerate करके हम CV में दिखा देते हैं, वैसे नहीं regeneration होता है. आपको 85% initial viability से जब drop होता है, when viability has dropped 85% below the initial viability, then only you regenerate. And you will be well aware that regeneration protocols are there where you have to follow for cross-pollinated and self-pollinated crops to ensure genetic integrity. And the number of seeds that has to go into the regeneration to ensure sufficient allelic diversity. For a crop like maize, when you have to go for chain sieving, or you have to ensure that you reconstitute to the population through regeneration and ensure the original allelic composition is there as in the base collection. आपको उतना chain sieving that the protocol has to be followed. For that, your initial population size should be there. अब regeneration के लिए you cannot pick out 10 seeds from the base collection and then go and regenerate and tell that, okay, I got back the sample with more germination. No. You should take care when you are conserving the sample. Regeneration does not just include improving the seed germination by just multiplying and taking up fresh seeds. It also means ensuring the genetic integrity of the sample by using the correct population size. Right? So that is how, just like that, you cannot take out and viability, put it in the field, 100 seeds, harvest and say that regeneration is done. No. Only when it is required, first of all, it should be done. When you are sure that, okay, individuals are getting lost, and many allele loss is going to happen, then only you go for regeneration. Otherwise, it is not required. It should not be done. You should maintain the base collection. Yes?
Ma'am, I missed the half an hour of your lecture because we had some trainee visit from the university.
No,
ma'am, another question.
We have enough time to discuss later also because you are here only.
Ma'am, one question. When we calculate intensity of dormancy, like we do in the groundnut, then ma'am, we have SOP not well defined. Like we do dormancy test in the Petri plate. And one, what we do is we do freshly dormancy, means freshly harvested pods are shelled and they are sown in the field. And one we do when in situ type of test, that is when pods are still attached to the plants. And then after the defined period, we can row out the plants and we can check whether the seeds have germinated or not. So ma'am, but there is a lot of difference between among these three methods. So what is the relation? How could that be worked out?
Groundnut, actually, to be honest, I don't have much experience working on groundnut. But I can look back and get back to you groundnut because we do laboratory seed testing protocols for dormancy breaking as recommended by ISTA. So before the planting, what we do is aapka paas seed lot when it comes for planting. Many times germplasm you do not get that much quantity of seed as in the case of the ISTA recommended seed lot and all. Aapka seed quantity may be limitation hoga. But even then, the sample from the germination, when you conduct the viability test, there are, thresholds we have tolerance limits beyond which, if there is a high level of fresh ungerminated seed, we recommend that that particular seed lots, fresh ungerminated seeds, have to be passed through the recommended dormancy breaking protocol. And what dormancy breaking protocol say, you will have to, and that should be done. And with that, if it is germinating, then that means the level of dormancy in that, आप उसी में से remarks में लिखते हैं, yes, this particular seed lot has this level of dormancy. And all the seed lot which is then going into sowing, which should follow that particular dormancy breaking protocol, and then only it should go into the field as a pre-sowing treatment. That is what the recommendation is. But in your case, I will have to discuss with somebody who's handling groundnut to understand, because I've never done this, what you have told, the field
Ma'am,
treatment
I
as
did
well.
experiment. I have what I have observed is there's a lot of differences. Means अगर in situ करते हैं तो different result आता है. अगर directly plants को pots छोड़ देते हैं फिर germination देखते हैं तो different result आता है. Petri plate में करते हैं तो फिर बहुत vary होता है result. So
this I will try to understand a little more from you in detail about what is the issue, and then discuss with you individually.
Okay, ma'am. Okay, sure, ma'am.
We'll
So much,
discuss, and I'll try to understand a little more about what exactly is the issue, because you'll be here only and it is an in-house discussion. We can do it anytime.
Okay, ma'am. Okay, ma'am. Okay. Thank you, ma'am.
Yeah.
Any other participants?
Hello, ma'am.
Please,
Yes,
can you?
Myself, Dr. Vinod Kumar from JNKV Powar Kheda, ma'am.
Yes, yes.
I am asking that any fungicide can enhance the germination percentage?
No, see, fungicide will enhance the germination percentage in the context that it will, so in the context of seed treatments, what it does is the primary infection level, when it regulates, it gives a higher, it is not going to enhance the germination potential of the seed. See, no chemical can enhance the germination potential of the seed, but the expression of germination gets facilitated by subsidence of the fungal attack.
Yes.
So what happens is, wherever there is a possibility of fungal infection, we recommend seed treatments like Captan in order to eliminate the presence of secondary and primary infection on the seed and occurrence of more normal seedlings. In that way, the planting value gets enhanced.
Okay, thank you, ma'am.
A seed that is dead, definitely nothing can be done.
Okay, thank you.
Ma'am, another participant, Dr. Gautam, ma'am,
Yes.
from ICARDA FLP, Amlaha,
Okay.
he is asking question, ma'am. Actually, in chat box it is.
Okay, sorry, I couldn't.
Yes.
Yeah,
I
I'll just see the chat box.
Yes, ma'am.
Yeah, I have problem in germination of soybean breeder seed after harvesting. We tested for germination, but we find that its germination is very low, around 50 to 60%. What may be the reason? So this is a freshly harvested soybean. Actually, it comes in the poor storer category, but I think now he's talking about freshly harvested. Problem in germination of breeder seed after harvesting immediately. So in soybean, in our experience also, imbibitional injury is a very serious issue. And second one is the kind of harvesting that is done. Like in mechanized harvesting, if it is a germplasm, in many cases, what happens is if in the case of improved varieties and all, that part is taken care of. But in the case of germplasm, since he's from ICARDA, I think germplasm may be his handling must be of germplasm also. What happens is the mechanical injury is also very high in soybean. And those seeds which are higher mechanical injury, and also even otherwise, the seeds which are susceptible to imbibitional injury is also very high. So when these seeds come in contact with free water in soybean, we can see that drastic loss of viability is there, even in the case of freshly harvested seeds. So these like the imbibitional, the pre-soaking at 100% RH, which we have recommended for soybean, that is one trial which can be done to see whether it is imbibitional injury. So even after doing that, if it is having the same problem continued, then you need to look into the mechanical injury that is possibly happening during harvest. So so we need to one by one rule out what is the cause. And after, if after harvest, if it is stored for certain duration, then because ICARDA definitely has the highest of facility, so I think these are not the storage issues. But basically, these are issues which we also face here in gene bank in the context of soybean, imbibitional injury. And some genotypes at lower temperature, it becomes more susceptible to imbibitional injury. So one by one, we need to look into what first thing I think what should be attempted is the imbibitional injury. And the pre-soaking at 100% RH at 20 degrees centigrade, and then taking the seeds to further germination should, I think, to a great extent solve.
And all seeds are low storers generally.
Yeah, but I think he's asking for freshly harvested. That is what I was thinking, because he's not mentioned storage.
Harvested freshly and then tested it.
Yeah, Dr. Amit. Yeah.
Yes, ma'am. And this
have you
this
tried
seed was
this soaking at 100%, not soaking actually, exposing to 100% RH for 1 or 2 days at 20 degrees centigrade?
no, madam, not yet.
Yeah, you can try that for 1 or 2 accessions and see the result, whether any difference is there. Because some genotypes are very prone to, so maybe your genotypes which are handling, they're very prone to imbibitional injury, means you can try that and see the difference in result, whether any significant difference is there. And then you can try, is it a mechanized harvesting or what is the mode of harvest?
Ma'am, some seed we have harvested through harvester, and some was by mechanical harvest. No, not mechanical, by hand.
Okay, any difference between these two also you please observe.
Okay, sure, definitely. Thank you.
Okay.
Another
question on white-seeded sesame dormancy is very prolonged than the black-seeded sesame. In sesame, what we have observed is that, see, we cannot categorize dormancy as, a case of white having more than black. We cannot have a flat rule that the seed color is strongly associated. There are contradicting reports on that. It is varying from cultivar to cultivar, from genotype to genotype. There are differences, but it cannot be that all white-seeded sesame will have a longer duration of dormancy than black-seeded. That cannot be. This must be the case of genotypic difference only, because we have come across contradicting performances in the case of sesame. So maybe the genotypes which the particular breeder is dealing in that it is so. But when you look into large number of accessions, you will definitely find even black-seeded ones which are equally having dormant nature as white-seeded, more than white-seeded also. So color is not the ultimate criteria.
And any other participants? Ma'am, I hope. And ma'am,
major problem is low germination in soybean varieties in Maharashtra. What are the reasons for low germination? So if this is pertaining to which are in general area in Maharashtra, when we are speaking soybean belt, in whichever soybean belt, it need not be Maharashtra alone, because soybean is a crop where viability issue is serious, and many of our seed certification and seed production systems are not equipped with proper storage, which is required for a crop like soybean. So after production, harvesting, handling, certification, coming to the certified seed production, and then finally coming till the sowing duration, sowing season, the conditions it undergoes, it's not congenial for maintenance of viability in a poor storer like soybean. So what happens is, wherever the gap is more, you can find a drastic drop. So we cannot afford to have that big that gap. We try to compensate it with a higher seed rate. That is how in soybean we try to manage. And mechanical injury is also more in field performance gets affected. In these varieties, the mechanical injury is a very big issue, because all these are mechanized harvesting in the certified seed production chain. So and that combined with unrecommended storage practices, because nowhere in India currently we can afford for certified seed storage, low temperature storage structures. So the first affected crop is soybean. Every stage grows through the same, but the first seed to give the indication of losing viability is soybean. So that gets visible first. A crop like wheat, where seed rate can easily compensate for even a small drop, and that drop doesn't become visible because of the seed rate, as well as the drop is not that predominant as in the case of a crop like soybean. And it is not visible in the field that much. In soybean field, it becomes more visible.
Ma'am.
Yes, please.
Ma'am, this is Dr. Dhanavendan, madam, from Central Silk Board.
Okay.
Silk
Yes,
Board. Sure.
madam. Ma'am, so far we are explaining maximum all cereal and this pulse crop seed. But in our case, like non-mulberry tree plantation is there, madam. In
Okay.
case of tree plantation like Arjuna and Kesuru, madam, that seed emergence, that seed germination very, very less than 25 or 30%, madam. That kind of tree plant, that is a perennial tree seeds, how we can improve the germination or how to that germination percentage will increase, madam. And what is the way to drive it or increasing the population or increasing the
in such tree species, in majority of the cases, you know, the identifying the exact harvest maturity stage, and number two is once the harvest maturity is finalized, you know, the level of dormancy. It can be a physiological dormancy because in tree species they are normally, you know, naturally evolved to have a certain level of dormancy because that is its survival mechanism under natural habitat conditions. So a certain level of physiological dormancy always prevails. So at times it is just getting over once it is a crop where a lot of breeding has gone into, this particular dormancy element is addressed. But in those species where breeding interventions have not happened and it is still, you know, you're relying on natural existing material, those genotypes will have a certain level of dormancy. So many of the tree species, this International Seed Testing Association has standardized the dormancy breaking protocol. And, you know, in tree species, viability testing itself will take a month to test it even in the laboratory conditions. In field, the emergence period will be still more also. But from the seed lot, the harvested seed lot, and another most important thing is when a tree species seed lot comes into you, very rarely you will get even 50% of the seeds in the same level of maturity. That is how the tree bearing happens, right, for seed production. So in many cases, majority of the seeds within the seed lot will be at a very low physiological maturity and it wouldn't have neared its germination period also. So you need to, you know, screen out and make the seed lot first, the quality wise at the same maturity level, identify the immature seeds and filter it out. And from the mature seed lot, you need to look into physiological dormancy. If there is an ISTA worked out protocol for that species, well and good. You can follow that for breaking dormancy and then testing in lab and taking only that particular seed lot to the field. And if it is not, you need to look into related species. Therefore, it has been standardized. Trial and error, apply the same protocol, and whatever clicks, you need to standardize. The standardization has to be done. So when you have 2-3 species of specific interest, you need to do that for that particular species and standardize it. And then most importantly, screen out immature seeds and you make your seed lot uniform with uniform maturity. And afterwards, you go for laboratory test. If physiological or physiological maturity, there is a cut test and then the viability testing you can do for that. That also requires standardization. You know, for doing the viable tetrazolium test also, you need to have basic protocol in place. How much preconditioning has to be done. So similar species where it has been standardized in other trees, you need to look into and apply and standardize in that and then go ahead with the actual field sowing.
Okay, thank you, ma'am.
Another question came in chat box.
What is effect of Selphos on germination? Yeah, which is used for fumigation against stored grain pest. See, again, as I told, this seed treatments which are being done, they are all physical interventions to eliminate damage to the seed, pathogen attack to the seed. Selphos will not have a negative effect if it is used in the recommended dosage. Higher dosage has toxicity on the seed. But the Selphos effect on germination is not a priming effect or anything. It is a physical effect of eliminating the infestation by stored grain pest, which is going to ensure the maintenance of viability by preventing the physical damage to the seed. It does not have any other hormonal or physiological impact on the seed.
Yes, ma'am. Any other questions? I hope, ma'am, there are no questions
Yeah, I can see that they are
Now,
all waiting for winding up.
Now it is a time for express to propose the vote of thanks. Ma'am, thank you very much for briefly discussing about the principles and procedures of viability, seed viability and seed germination, and sharing your knowledge and minimal practical basic principles. Nicely, you expressed in a very nice manner, and it is very highly useful to the participants. And I saw very much interested participants also asked the questions. Very nice, and they interacted with you. And thank you very much, ma'am. And thank you, Harish sir. And thank you, participants. And please switch on videos. We will take one screenshot. All participants are requested. Vijay Mandal ji.
Yes sir, 68 participant है, अभी लोगों ने नहीं किया है on.
Yes, yes.
अभी काफी लोगों ने वीडियो ऑन नहीं किया है.
Somebody using Zoom is actually, please kindly mention your name and institute.
Sir, अभी लगभग 30 जनों ने ऑन नहीं किया है. कैसे करेंगे?
Dr. Rakesh Chaudhary, Dr. S. Maheswaramma, Dr. Muhammad Maqbul Rathar, Dr. K.N. Gupta, Dr. Monica Singh, विजय मंडल जी.
यस सर.
हाँ यस, आप ले लीजिए, जो स्विच ऑन किया वो ले लीजिए.
ठीक है सर, हो गया.
हाँ ठीक है. Thank you, Vijay Mandal ji. Ma'am, thank you, ma'am.
Okay, thank you.
Harish sir. Thank you very much. And
thank you all of you.
Thank you, dear participants. We will join tomorrow at 9:30, and tomorrow sessions are very highly useful. And the morning session is management and utilization of genetic resources of cereal crops. And after that, registration of trait specific germplasm is there, and afternoon practical is there. And tomorrow we will meet at 9:30 a.m. Have a nice day. Thank you. Thank you very much.